as well. Okay, good afternoon, everybody. Thank you for joining us for this early career conference, pre-conference symposium. Let me also do the RMS's first ME. I'm glad that we can all make it here today. Let me just introduce myself. So my name's Liam Rooney. I'm the chair of the Early Career Committee for the RMS. And together with the committee, we have designed today's programme around platforming some of our early career members. So we've got a really packed in schedule. So without further ado, I'll give a quick overview and some logistic things to point out as well. So for those of you who require, there's closed captions are live during this. So you can click at the bottom of your Zoom screen if you require them, and it will turn on. I really apologise for my Scottish accent in case anything weird comes up. I can't help that, I'm sorry. And then I really just need to thank, first of all of our organisers, our speakers and our chairs for today, and to our affiliated sponsors for helping us get this meeting on track. So as I say, we've broken this into kind of two sections. So our first section has some contributed talks, followed by a keynote speaker, who's Dr. Michel Conroy from Imperial College. And we will then swiftly move on to our panel session, and I'll have some more words to say on that in a minute or two. We'll then have a quick comfort break before we then move on to some more science, some more and we will round off the afternoon with our RMS Early Career Award lecture, which has been given by Dr. Kevin Whitley from Newcastle. Kevin was the recipient of the first RMS Early Career Award, so we're really excited to see his talk. I'll then close the meeting with the section AGM, which is our first annual section, and I'll kind of just open the floor at that point if there's any kind of feedback or comments that anybody has for the Early Career Committee and how we can do a better job of representing the community. So a quick note on how the panel discussion will work. So we've got a diverse number of panelists here from all different areas of microscopy, and we'll be concentrating on two main themes of career pathways and of securing funding as an early career researcher. The panel discussion will be facilitated via Padlet. So this screen here, if Rhiannon is on the call, if she could drop a link to this in the chat. We'll drop another one later, closer to the time. But this is essentially a portal which will allow you to post anonymous questions to the panel. And you can do so by just kind of double clicking on the white space and write your question to the panel. This will be shared live and we'll update live for our panelists to see during the call at that point, and we can take it from there. If there's any problems with that, we'll kind of tackle it as we go. I think we've tested it, hopefully it works, fingers crossed. I've got a couple of exciting announcements to make. The first is that as part of our programme, we have a prize for the best contributed oral presentation. So that's for our 4 contributed oral talks. So the best of that will receive a prize, which is sponsored by the Early Career Committee. And the second announcement is just to kind of emphasize that after this meeting is over, you've got the rest of MMC to go to. So if you've not registered for that, then there's still an opportunity. And there's also things like the exhibition and the trade show, which are completely free, that I encourage you to go to. If you're not presenting, can I ask you to turn off your mic as well? That would be a great help and reduce background. Thank you. The second and third announcements are some exciting ventures that the committee has with the Journal of Microscopy. So the journal is launching an open call specifically for early career researchers. And the point of this is to bring to the forefront of recent microscopy developments by early career researchers. And submissions are open from every field of microscopy for this. The guest editors for this issue will be myself and Dr. Laura Clark. And if there's any kind of informal inquiries before this launches, then feel free to get in touch with us. Keep an eye on your inboxes over the next couple of weeks, and we'll send you out some details of when this officially launches. And for the Electron microscopists out there, some exciting news for December coming. The Society for Electron Microscope Technology is having its meeting in December, and that will also feature the RMS specifically for students, interns, apprentices, and faculty staff to present their research at a high impact meeting. And there's a prestigious award associated with that as well. So please keep those dates in your diary. Now, without further ado, let me stop talking and hand over to our first chair for this first session, who's Dr. Heba Salim from Oxford University. So Heba, if you want to take it away, that would be great. I'll stop sharing, and thank you very much.
Thank you, Liam. Welcome everyone to our first session, and we have two contributed talks. So we will start with a recorded talk from Dr. Michel. Sorry, can you hear me?
Yeah, we can hear you. Yeah.
Sorry, it will be good if anyone can mute if they are not presenting. And we will have the first talk from Michel Budek from ETH Zurich University, and we will have live questions at the end of that. So Michel will talk about a new technique, scanning field emission microscopy using a miniature bezel box electron energy analyzer. Michel, please feel free to start.
Hello everyone, my name is Michel Budek, and today I will present you my work concerning scanning field emission microscopy and implementation of bezel box electron energy analyzer into this technique. First, I will briefly introduce what is the scanning field emission microscopy. Then I will tell you what is the golden standard of electron energy analyzing. And in the end, I will present you the bezel box energy analyzer, what are the advantages, how does it work, and what kind of results we obtained using this detector. Scanning tunneling microscopy is a pretty established technique. We have a very sharp tip that is brought close to the sample surface, and we put a low voltage between the tip and the sample, typically around like 1 volt, and we are measuring the tunneling current between the tip and the sample. And scanning field emission microscopy uses the same instrument, but we retract the tip to larger tip sample distance, typically a few nanometers up to maybe a few microns, and apply a larger voltage between tip and sample, typically tens of volts, maybe a few hundred volts, depending on the tip sample distance. This way we will create a field emitted current originating from the tip that will be pushed by the electric field into the direction of the sample, and we can again detect the absorbed current on the sample from this electron beam. It was previously shown that the resolution of such microscope is around 1 nanometer. Here you can see for comparison images of scanning tunneling microscope and scanning field emission microscope on the same sample. But on top of the microscopy, we can use these secondary electrons that are scattered from the sample surface and use them for an electron spectroscopy measurement. But for that, we need some kind of electron energy analyzer. And the golden standard is a hemispheric analyzer that is used, I think, in all XPS chambers. Here we can see an image of such an XPS instrument where this is the measurement chamber and this is the electron energy analyzer. This analyzer works that we have a field that is causing a curvature of the electron trajectory, and the different energy of electrons will have a different radius of the curvature, so therefore we can distinguish between different energies of electrons. But the biggest disadvantage of this analyzer is its size. So we can see even here that the chamber and the hemispherical analyzer are approximately the same volume, so we need to maintain a UHV in approximately twice the volume when using the hemispherical analyzer. This is a picture from our setup, and we really see that our hemispherical analyzer is approximately as large as the whole chamber itself. So this consumes a lot of energy to maintain vacuum in large volume. So what if we can use something smaller to detect energy of electrons, and we use so-called vessel box that is using a retarding field to distinguish between electrons with different energy, and it's really a miniature design that is only a few centimeters large. You can see that really the length of such an electron detector is only 5.5 centimeters approximately. And the composition of the vessel box is like this. We have first an entrance cone with an entrance pinhole through which the electrons enter the vessel box. Then we have the main body of vessel box, and in the end we have a second pinhole, the exit pinhole, through which the electrons enter the channeltron that simply counts how many electrons we are obtaining. And how does it work? So this is the cross-section of the vessel box, and let's say we have an electron beam with energy of electrons, let's say 50 electron volts, and they enter through the enter pinhole. And if we set minus 50 volts here on these vessel plates, the electric field here will cause that the electrons that are coming under some kind of angle here will be bended. And the geometry is constructed in the way that the electrons with energy corresponding to the voltage on this vessel plate will get nicely focused into the exit pinhole, and they get counted here on the channeltron. If we send electrons with lower energy, so let's say 45 or 40 electron volts, then the electric field from this vessel plate will cause them to bend, and they will be repelled back to the front cone of the vessel box, and they will not enter the exit pinhole, and they will not be counted. And if we send their electrons with higher energy, so let's say 100 electron volts, then the electrons that have the trajectory more or less parallel with the axis of the vessel box, they will get through the exit pinhole because the electric field is not strong enough to repel them, and we will count only few electrons here. But the electrons that go under an angle into the vessel box will have not enough time to bend and enter the exit pinhole, so we will really detect only a negligible amount of electrons here compared to the focused electrons.
Oh, yeah.
This way, we can create a very small electron energy analyzer that has an electron energy resolution around 1 electron volt. The second big advantage that comes with the size of the detector is that we can implement it into the measurement chamber without the need of increasing the volume, and we can mount it into motors or micro screws, and we can have 3 degrees of freedom of movement of such a detector, so it can move along the sample, perpendicular to the sample, and up and down. And this way we can really focus to from where we want to collect electrons. And this is just an example. If we are moving the vessel box along the sample, we can really always find an optimum position for the measurement where we have the highest number of counts. So this was how it works, and now let's see what are the results. So these are the measurements on the clean tungsten 110 surface, measured with Bessel box and hemispherical analyzer, and we can see in both cases there is a strong peak of elastically scattered electrons with 25 electron volts energy. And in our case, even the Bessel box has a better energy resolution than the hemispherical analyzer. And the spectrum of one and the second detector corresponds pretty well. If we change the material, this is cesium deposited on this tungsten 110 surface, and we can see in Bessel box that we can already distinguish between two peaks. And in hemispherical analyzer, because of the lower electron energy resolution, we cannot really distinguish the first and the second peak, but when we compare this spectrum to the spectrum of clean tungsten, it's pretty clear that here is also hidden a second peak. So to conclude, we incorporated a miniature energy analyzer, so-called Bessel box, inside our chamber. The biggest advantage is, of course, the size and the mobility of such a detector, because it has adjustable working conditions. We can move it around, we can get it closer to the sample or further away from the sample. And also, when playing with the geometry of the Bessel box, we can change the resolution, or basically the area from which the electrons are counted. And we show that the energy resolution is around 1 electron volt of such a detector in our setup. In the end, I would just like to thank all my colleagues from Zurich or from York that helped this work to work. And of course, I would like to thank you for your attention.
Thank you very much, Michael, for interesting talk. Okay, so while waiting, okay, there is a question from the audience from Liam. Are you able to hear me, Michael?
Yes, I can hear you.
Okay, good. So the question is, with the adjustable position of the box, how different is this between different samples? Does it require a lot of optimization?
Yeah, this is one thing that, of course, when we change the sample or when we change the tip, the position or the spot from which the electrons are actually emitted differs. So we need to move the Bessel box always if we change the tip or the sample. But actually, it takes maybe half an hour or 1 hour to really align it to the best possible position when we change something inside. So it takes a bit of work, but it's not that hard to find a new position.
Okay, so that's take me to my question, like, with this technology, what do you think is the maybe some limitations with the proposed technology, or is it, do you see like there is specific application that is more suited with this technology than others? Like, can you elaborate on that?
Yeah, I think the biggest difference from the standard energy analyzers are the range where we can measure, because this kind of detector is quite limited to the highest energy we can detect, because otherwise when we send few, I don't know, kilo electron volts electrons, they will just pass through because they don't have enough time to actually bend. So for the applications where we need really higher electron energies, then I would say that this design would not be actually optimal. And the standard energy analyzers would be better. But in the applications where there are not kilo electron volts, maybe few hundreds of electron volts of electrons, then I think this is pretty usable.
Okay, okay, great. Okay, thank you very much. So in interest of time, we will move to the next talk. So our next talk is from Gan van de Kerkhoff from University of Cambridge. And she will tell us about nutrient-induced changes in the cellular organization of structurally colored bacteria colonies. Gia, please, the floor is yours. Thank you.
Hi, I can't seem to start my video. It says the host has stopped it. I can present without the
Can you
video,
share your
but,
screen? I can't see your screen yet.
Yes. Can you see it now?
Yes, yeah.
Okay. Also in the presenter mode, or is it now?
It is in, no, if you can
Better?
switch. Yeah, yeah.
Okay, good. All right.
Great.
So my name is Gea, and I work on structurally colored bacteria. Structural color is color that doesn't come from pigmentation, but instead it comes from the interaction of light with structures at the nanoscale. So perhaps the most famous example is this butterfly that you see here. It's called the morpho butterfly. And the blue in the wings of this butterfly doesn't come from pigments, but it comes from these tree-like structures in the scales of its wings that create an interference mechanism with the light, which gives it a blue appearance. This kind of coloration is quite widespread in nature. It occurs in animals, but also in plants, like this pollia fruit you see here, where the nanostructures are very different from the butterflies. It has these arced layers. But it also occurs in bacteria, where in bacteria, this kind of coloration comes from the organization of the cells within a colony. And this is quite an interesting concept because the organization of the cells is directly linked to its optical response. We can therefore use the optical response to characterize the behavior of the cells in the colony in vivo. So what we have been working on is to study these kind of systems, and to do that, we first developed a method to sort of derive the cellular behavior from the optical properties. A start on that is to just resolve the sort of basic nanostructure that's responsible for these colors. So if we take a colony on a petri dish, on agar like here, and we image it with cryogenic SEM, if we look at them on top view, you see these rod-shaped cells here that are lined up quite nicely next to each other. And if we then look at that in cross-section, you see the image on the right here, where we see the cells packed in a sort of hexagonal lattice. And it is actually this hexagonal packing that gives the colony a crystalline structure, which is a photonic crystal, which is in the end what gives it its colorful appearance. So EM is very nice here to resolve the basic structure, but you can also think about details in this cellular organization within the framework of this hexagonally packed crystal. And what I mean by that is basically the level of ordering within this colony. And we've identified 3 different levels of that. The first one that we're going to look at is the local ordering or disordering, where in a perfectly ordered hexagonally packed system, the cells will give a really high intensity color because the structure is really nice. But if there's a certain level of disorder, then this intensity will decrease until the point where the structure is completely destroyed and the color is gone. The second level where we can think of is because these structures have a direction, the light that is reflected from it will also be directional, meaning that it will be angular dependent. Now, if within such a large structure there is a small subunit that is tilted, then the angle at which this light is reflected will also change. So depending on how much these structures are tilted or not, you can get more matte or more iridescent samples. The third part is that we can also think about the lattice constant or the interbacterial distance of the system, which is a parameter that directly relates to the wavelength of the reflected light. Now, these different levels of ordering within such a colony they can all be derived from the optical appearance of the colony. And what we've done, we use this system of Flavobacterium IR1, and we subjected them to 2 different nutrient conditions, one of which we get a iridescent green appearance, and the other one where we get a more matte purple and slightly less intense appearance. The first thing we did is to look at them with light microscopy, just in dark field, where you see the 2 different nutrient conditions here. And what you can see is that on a one condition, we see this really nice green, and it's kind of present everywhere. But in these other nutrient conditions, we see that there are locally these black gaps in the colony. Now we know that this is not caused by an absence of cells. There's no holes in the colony. And we know that from cryo-SEM, where we look at them in top view, and there is a continuous film. So there's not that there's an absence of cells there, which means that there are local domains in this specific system with this added fucoidin to the nutrient mixture, where the cells must be too disordered to give a color. So we can use the microscopic features in the optical appearance to study this kind of disorder. Then, like I said, this color is angular dependent. We see here a colony of the IR1 bacteria on agar, which is mounted on a glass slide. And you can see that in some angles, the color appears, and in others, it's not visible. And we can study this angular dependent color with a goniometer. So what we do is we use this setup here, where we keep the incident light angle constant on the sample, and then we measure the light that's reflected from it for a whole range of different angles. And what we get is a plot like this, where we see the reflectance spots here and here, resolved for the wavelength here, and the detection angle here. And this is quite important because the exact angle where which wavelengths of light are reflected are very crucial in the analysis of the microstructure. Where if we were thinking back about these tilted domains, which I was talking about earlier, like here, where you have, if there's a crystalline domain that's tilted, the light will be reflected on a slightly different angle than from when it's not tilted. Now, we have, for the standard condition here, we have 2 main peaks, and we assume that the main peak will be caused by the situation in number 1 here, where all the bacteria are aligned to the surface of the agar, because that will be the standard configuration for them. It's the easiest way for them to organize. However, if you look across these intensity peaks here, you can also see a bit more clearly here, there is an angular streak to this optical signal. And this angular streak that's actually caused by these tilted domains, which means that the stronger this angular streak is compared to the main peak, the more of these tilted domains you'll have within the sample. And we can, so this is the aligned domains, this is the tilt domains. We can quantify that a little bit to compare these different systems by taking the intensity of the aligned domains and compare it to the tilted domains. And the ratio between those two will give us an idea of how many tilted domains there are in the sample, where we see that the fucoidin case, where there's more matte appearance, will also have more tilted domains. Then lastly, like I said, we can also look at this intercellular distance between the bacteria. And the way we can do that is because these bacteria colonies, they also act basically as diffraction gratings. And because they do act as diffraction gratings, they follow this grating equation, which links the interbacterial distance, this parameter, to the wavelength and angle at which certain light is reflected for a given incident angle. So what we do is we can fit this equation to these reflection spots, and we can get the interbacterial distance for the 2 different colonies. And we see that the difference between these 2 circumstances is only 25 nanometers, but we can still detect it. And as a matter of fact, when comparing different measurements, we found that we can actually use this method to get this lattice constant with a resolution of 10 nanometers, which is really nice for studying these kind of behaviors within a colony in vivo. So to summarize a little bit, we have these structurally colored bacteria colonies that are a very unique system to study the cellular organization within a living colony without doing any kind of sample prep, and we don't have to disturb the cells in that sense. And we can get both large area and local behavior from this. And we can determine the intercellular distance with an accuracy of 10 nanometers, which is really high. Yeah, I would like to thank the people from my research group, specifically the ones that have been working with me on this system, and also the people from Hoekpijn, which is a company in the Netherlands that works, it's a biotech company that works on structurally colored bacteria. Thank you very much. I will take any questions if there are any.
Thank you very much, Gia, for very interesting talk. While the questions coming in, I will ask a question. How do you think this can be used in like a drug discovery context, or like can you see this used in high throughput fashion to characterize different like nutrients effect on biological systems? And I have requested from Liam to turn your camera on if possible, please.
Yeah, I have, I think. Can you not see me?
Okay. Maybe because you're sharing your screen, I can't see.
I can stop sharing my screen if that's helpful. Yeah, so these bacteria, they are very promising as sensors, in the sense that the nutrients that I showed here, there were the difference between the different conditions was that the one just had a regular mixture, and the other one is basically the exact same mixture with the addition of this polysaccharide. And we see this massive change in the appearance. And so this is for a very specific compound now, but we've actually looked a little bit into the genetics of this system. And it's a very specific genetic pathway that's being used for this one polysaccharide, which makes it a very promising system in the sense that we could potentially in the future modify the other pathways to become equally sensitive to different types of compounds. So yeah, I definitely think there's some potential there.
Very interesting. So what do you think is the functional value? Did you have any thought about what the functional value of having such different patterns, or are they attracting different things by this behavior?
That's a very good question. I think this is the big question that we have on this system, and I don't fully know. So we recently, there's recently been a paper where people have found a function of it, but it's not related actually directly to the optics. In that specific case, they found that it's the organization of the cells that helps as a predation mechanism for other microorganisms. So basically, when the bacteria are low on food, they will use this kind of organization to sort of start hunting other bacteria. But that is not to say that that is necessarily the only mechanism or the only reason that they do it. It's just the only one we have found so far.
Cool.
I can speculate for a long time about possible other mechanisms, but
okay, cool. Okay, there is another question from Kevin. Is there any way to predict the wavelength versus angle results, and such based on the SEM images or diffraction equations?
To some extent. So in principle, there's a few factors here. So if you assume that the refractive index of the system, if we know that, because it's another aspect that I haven't talked about yet, but if you assume that that's always the same, then in principle, if you can just measure the interbacterial distance from SEM images, you can get a prediction for the wavelength that's being reflected. That said, we have tried this with SEM, but because you are looking at such a local area, and because there's always slight artifacts from sample prep and then the orientation of the cells in the image, we found that there in the end we get much more accurate results by just looking at the optical response and not looking at the images in that sense. But yeah, you can definitely get an approximation. Yeah.
Okay, for one last question, go quickly from Liam. Is this gliding species of flavobacteria? If so, how does the gliding organization affect structural color?
Yeah, it is a gliding species. It's very closely related to the Johnsonia. So we know that the gliding behavior does affect the structural color, because mutants that have knockouts in the gliding, they are much less effective in the coloration. So it's definitely a factor. How exactly, I'm not sure, beyond the fact that it is a factor in that sense. Yeah.
Okay, thank you very much for your talk, and we will move to the next talk in the interest of time. So next we have our, thank you. So we have now our keynote speaker, Dr. Michel Conroy from Imperial College London, and she will talk about probing the dynamics of charge topologies with electron beam. Just a brief overview from Dr. Michel is a Royal Society University Research Fellow in the Department of Materials and at Imperial College London. She specialized in high resolution in situ STEM and EELS, and she worked at the Pacific Northwest National Laboratory as a TIM staff scientist before joining the University of Limerick as a Science Foundation Ireland Industry Research Fellow. In collaboration with analog devices, she investigated changes in physical and magnetic structure of ferromagnetic domain, wall-based devices by differential phase contrast STEM. So I will not elaborate more. I will, without further ado, I will leave the floor to Dr. Michel. Please share your screen and when you are ready.
Thanks. Okay, just if everyone, can you see that?
Yeah.
Yeah, okay, and I'll put it in presentation mode just to make, is that okay?
Yeah, great.
Great, okay, thank you so much for the introduction. Also, thank you so much to the Royal Microscopical Society, more specifically the Early Career Researcher Committee as well for inviting me. So today I hope that I can convince you that we can use the STEM probe and not just avoid it for that. For I guess some people think these are negative effects from the STEM probe. So there's lots of different reasons why there's a benefit to using a STEM probe, and more specifically an aberration corrected STEM probe, because if we're trying to analyze something at, let's say, the subatomic scale, what better way to do it than to probe it with something that is at that subatomic scale also. So today I'm going to talk about charge topologies, and what that basically means is, in this case, these are ferroelectric domain walls or vortexes, and the overarching naming of that is topologies. And I want to be able to switch them from a charged state, so let's say conducting or more insulating, to then a neutral state and back again, switching back and forth without ever harming the material itself. So we're truly trying to understand what is happening at that subatomic scale. So I started this research at the University of Limerick within the Burnell Institute. And Professor Ishi Bangert has her group there, her TEM group, and there's some talks this week as well with MMC from the group as well. So if anyone wants to check those out. And so a lot of the people in the group focus on in situ microscopy, again, not just using in situ holders, but also using the STEM probe to move different atoms around. And then also some examples of this. So doing liquid cell work and gas, heating and biasing. So this is where I started off doing this ferroelectric domain wall research. And then more recently, I've moved on to Imperial College London. And here we have a specialized setup. And if anyone has any interest in this, and I'll explain it in a second, please reach out. So we just got some of our microscopes set up and signed off last week. So we're very excited. So this is where we can make our samples, and let's say in a focused ion beam, in the SEM, all under cryogenic if you want to, then transfer them to the TEM in this case, and then over to the atom probe as well. So you get this whole cohesive trying to understand what maybe in some material systems that have lighter elements, or something where that material, let's say, that is lithium or hydrogen would just evaporate out under normal conditions. So this is the new setup, and it's a 40% access to other EPSRC grant holders as well. So it truly is an open access thing. So again, contact me if you need any more information. So what I wanted to do to start off this talk was to talk about my career pathway, because mine's not necessarily the most direct. And I think sometimes we're told, I know I was told, oh, you need to do so many, let's say, postdocs, and you need to apply for certain grants. And if you don't do that, you might as well give up, and all this kind of negativity. But mine definitely wasn't the traditional direct route. So when I started my PhD in Ireland, we didn't have an aberration corrected TEM. So I did a lot of internships in different places. And then at the end of my PhD, I decided I really, really love TEM, and that's what I want to focus on. So I applied for a postdoc at Pacific Northwest National Lab, and that's over in Washington State. And why I liked doing my postdoc there was that I could completely focus on TEM and then really upskill myself in the microscopy techniques that I liked, that I was really interested in. And then after that, I was promoted to staff scientist. That's also a nice benefit of doing a postdoc at a national lab. There's always that kind of different route instead of going the academic route. And then I took a career break for family reasons, and I moved back to Ireland. And I was very lucky that at that time that we got 2 aberration corrected microscopes, so one in Trinity College Dublin and one in the University of Limerick. I joined the team under Professor Ishi Bangert, and there then I kind of wanted to get going again on my own little kind of carved out research career. So there's a really nice funding scheme called an Sfi Industry Grant, and that's open to anyone around the world as well, and with any company. So if anyone wants to look that up, and that gives you the ability that if you're that kind of quasi between being a postdoc and being perhaps a full-fledged PI as well, you're given either a year of a grant or 2 years part-time. So you could continue doing your other kind of postdoc work, but where you are the PI on the grant. So you're not affiliated with another PI. And that gives you that time of independence. That's how I felt anyway. And then what's nice is for me, I got to get into the world of magnetics as well, because I was so focused on all types of electronics, but really I had not gone into the magnetic field. So analog devices gave me that. And then I was able to look at real world devices and work with the R&D group and use aberration corrected microscopy to analyze some questions that the fab had that we couldn't have figured out, because these devices really were there was only 1 or 2 atomic layers for the magnetic domain wall devices. So you need TEM. That's a really great example of why you need TEM in industry. And so then when I was doing my fellowship at Analog Devices and UL, then I applied for the Royal Society grant. And I was very lucky to get that. And so for the next, that kind of sets you up more properly to go down your own route and set up your own group. So that's what I'm doing at the moment. I hope that helps anyone. Okay, so then what this talk is about is why ferroelectric domain walls / topologies. So why I love these, that I started off with quantum wells in 3 nitrides, and I loved all electronic materials. But the thing that I was looking at, although it produced beautiful light, I could not move it. So once I form the device, I can't change that after in post-processing. I can understand maybe why it failed. I can understand maybe where I went wrong in the growth, but I really can't change it after that. My quantum wells are where they are and they're not moving. But with multiferroics and ferroelectrics in this case, you can move the thing that's interesting, the thing that has that interesting property, whether it is a change in conductivity, or if it's multiferroic, perhaps it also has a change in a magnetic moment, so a spin, or it also has a change in band gap or anything like this. So there's lots of different interesting properties, but the thing that you're trying to look at can also be moved. So I like the analogy that these kind of topologies, think of long-term, that us, the end user, an Etch-a-Sketch, and that's our, and as the end user, we can change our device and we can change all the networks of, let's say, our wires or LEDs. And then once we change those, we can wipe them away, draw them again, or we can even just move them as well. So that's why I got excited about it. And the second thing is I'm a microscopist, and more specifically, someone that loves to look at atoms and subatomic kind of resolution as well. And so what governs, in this case for ferroelectrics, the reason why something becomes conducting or not, is that just a slight shift within the unit cell. So these slight shifts make all the properties that we're interested in. So really, again, you need aberration-corrected STEM. And my talk today is going to be focused on moving the domain walls and all these type of topologies with the STEM probe. So the STEM probe is not just used for the imaging, it's also used as the thing to probe what we're looking at. Okay, so where all this came from, I was saying before, I had joined the group with Ushi Bangert at UL, and she had an Sfi US Ireland grant. And really it's Professor Marty Gregg up in Queen's University Belfast, looking at the material by Roger Watmore, who is in Imperial College London, and where they were saying, well, why not, instead of looking at these traditional ferroelectrics, why not look at something where the ferroelectricity is actually governed by something else? So let's say strain. And then maybe the energy to form these highly charged regions will be much lowered because we're not actually focusing on the ferroelectricity. And they were right. And so they were able to produce these beautiful networks of network of these charged regions, and they were able to move them. And then they could show from a different type of microscopy technique, so conducting AFM, the change in conductivity there as well. So they were able to form these, but what I think is really exciting is that they were able to move them so freely. So on the left video, you have a light microscope video, and then that shows where Joseph Guy in that paper below there was able to move the domain wall back and forth and back and forth. And he actually showed also that it wasn't behaving in the way other classic ferroelectrics behave. But I'll get into that in a second. But what was really fun as well as an electron microscopist is when I took the bulk crystal and put it in the SEM purely to make my lovely TEM lamella, I could also see that the those charged regions, so these domain walls, were also moving under the SEM probe. And what I had done before my postdoc was I used the electron beam to manipulate things in different types of liquids in situ holders. So I started thinking, actually, maybe the SEM beam is doing something here, and this isn't just a weird kind of charging effect, especially when I saw that it was cyclical. So I kept playing with this. Then I moved on, and I took my lamellas out. And then I was able to see also under normal STEM HADF, so under normal STEM imaging, that I could also see that pattern changing. But I couldn't quantify that. So I changed to what is called STEM DPC. So simply, I have a segmented detector. So I take my ring detector, and in those segments, I see where the electron beam, after it's gone through my material, deflects onto. So does it go north, south, east, west, or does it kind of go between those two segments? And then I could see really clearly that all those patterns were changing. You can form these beautiful videos, but they do mean something. So what I like about the low resolution is that you can get this large field of view. So your fib lamella in that case is the entire crystal. It's the entire thin film. And then you can pick out the regions of interest. So are you looking at a conducting wall? Are you looking at an insulating wall? Are you looking at different types of junctions? Then you can go down to the atomic scale. So then you can move down one more scale. You can say that's the domain wall I want to look at. And then you can keep rotating it. So to understand what's happening. And by rotating, I mean the electron beam coming in scanning, because of course you can't rotate your sample once it's in a normal holder. And so you say, okay, that's the domain wall I want to go look at atomically. So let's go atomic. So we can do all these beautiful images atomically, which I will show in a second. But if we can't quantify that, we're just getting beautiful images. And I want to highlight some of the great PhD work by Dr. Owen O'Connell and his TEMUL toolkit to quantify that movements of the atomic positions is up on GitHub. So if you look up TEMUL toolkit, and this is where then we're able to take some of the work he had already done previously on 2D materials and then dragged Owen into the ferroelectrics world. So now we're able to advance that code and look at different types of unit cells as well. Okay, so we started off with this weird material, but just to briefly go into what does that look like atomically. So it's an improper ferroelectric. So as I was saying before, something else governs the ferroelectricity. So in this case, you take your unit cell And this is a ferroelectric. So the unit cell is squashed, and that squashing, which is the shear strain, results in a displacement of the atoms within the unit cell. And then that displacement results in a dipole moment. And then so that results then in the ferroelectricity in this case. And then depending what type of polarization or what type of two different domains come and meet each other, then you can get a conducting or an insulating wall or anything like this. But if you can't quantify that displacement, then you can't actually understand what's happening and what type of domain wall you have. Okay, and then sorry, the slide. Yep. Okay, so then at the start, we didn't even know what type of domain walls or materials we had, or sorry, what type of unit cell we had, because no one had actually a SIF file or anything like this. So we started right back at the start, and then we did DFT calculations with Sinead Griffin at Berkeley. And the wonderful thing is that if you're doing atomic scale imaging, then you can correlate directly to DFT. That's the great thing. And she was able to tell me, if those atoms move in this direction, that's your polarization direction. So we worked together looking at the experiment, making a SIF file out of the XRD data, and then relating that back to what we're getting experimentally in the microscope. So luckily enough, in my case, it was the heavy elements that were governing the polarization. Thank goodness. So, okay, so then we get to the atomic scale. And so this is some of the work I was doing with Lewis Jones at TCD and Clive Downing at TCD. So this is where we started off looking at a charged domain wall and moving it back and forth into this kind of less charged state to charged state. And then we're able to then go in, make our domain wall more stable, and I can go into that in questions how I do that. And then we were able to actually image the charged domain walls themselves, and we're able to say how thick that wall is, how rough that wall is, what's happening there. We're able to do ABF imaging, so we can look at the lighter columns as well. I would say with this case, because it's a very complex unit cell, probably better long term, and that's what we started doing, is doing IDPC imaging, just because it's picking up some of the lighter columns better, because it's boron and oxygen. And that was great. So now we can understand, okay, the charge wall is relating straight back to what we were hoping with theory. And then we can also do this. So this is another paper with Kalani, myself, and Ushi, where you don't always have to maybe go down to the atomic scale. As long as you get a nice FFT map, you can start mapping out your polarization with, depending, if you know the superlattice spots you need to look at, or the tetragonality, let's say, in some other types of ferroelectric materials. You don't always have to go to the atomic scale if you don't know what's happening at the start. And again, then we could clearly show that we can map this out, this kind of movement back and forth. So then going back to this, at the end of the day, it's still a ferroelastic, so we need to understand the strain. So we moved to 40 STEM, and I'll briefly go through this because I'm probably talking too long. But in this case, why we did 40 STEM was to look at the strain. And what I think is really exciting about this, and this is working with Colin Ofus at Lawrence Berkeley National Lab, and we were able to watch and quantify the shear strain change of different types of charge wall as they were moving. So I guess you could kind of say it's 5D STEM, because you're also watching the time evolution as well of this. But what we could also analyze from this is that there's this long range reordering of the shear strain. So it really is the shear strain that's, in our case, governing these highly charged states that normally would energetically not be favorable. It's great that we could see that now. And then this is the last thing that I really wanted to highlight, because this is the thing I'm trying to push at the moment. And we don't just move, let's say, domain walls around from this kind of state of 0, 1, 0, 1, charged, not charged, but also form different types of these really interesting topologies. So in this case, a vertex pattern. And again, so we go from two neutral walls. And then if I bring the domain wall, or if I bring the STEM probe in a certain direction, I can form these completely highly charged vertex patterns. And I've also started showing I can form these like vortex patterns as well, depending on what direction you bring the STEM probe in. So now we can start studying these kind of quantum entities that we couldn't really have done at that time resolution, at that spatial resolution before. It's not just this material, it's not just a once off. Across all these type of borosite materials, I'm able to move these topologies. So this is kind of a low mag image of the different domains moving around. But again, I've done this at atomic scale for 6 different types of borosites now. And hopefully it goes on to the next one. And then we're also looking at different types of material systems completely. And actually a really interesting thing that we saw, hopefully in the slide before, a really interesting thing that we could see was that in these kind of classic systems, what we thought were classic domain walls actually were forming anti-ferroelectric domain walls. And if you don't have any experience in any of that, what's really cool is that at every single unit cell there's a flip in the polarization. But if we'd never been at that resolution, we would have never been able to see that because there was no other technique that we could use. And then that matched up perfectly with DFT, but DFT couldn't have predicted this because no one knew that type of domain wall even existed. So this is where a TM was really needed. So the summary of highlights, the affected electron beam can be used, not just avoided. And this is in lots of other different types of applications. But I think heading into more quantum research, it really can be utilized. And we needed the resolution of, in this case, aberration corrected stem and the code development as well, because we have these vast quantities of data and beautiful images, but if you can't quantify what's happening with the atomic displacements, then there's no point to any of this. So really code development is very important. And of course, you can do all this in situ biasing and heating as well. On top of that, not just using the stem probe, and that's what we're also doing. And also we're using eels. So doing all this in the dynamics, so switching on and off, and then trying to see if there's any change in the band structure, band gap, and everything like that. So the acknowledgements, because I wouldn't be able to do this obviously all by myself. And I just wanted to thank our Colin Ophis and Jim Siston at Berkeley National Lab, and also Sinead Griffin, because without theory, again, we have lovely experimental data, but we need to back that up and also work properly collaboratively together. And Professor Quinton Renas at SuperStem for really probing into the low loss yields and all the band structure analysis, Dr. Lewis Jones and Clive Downing at TCD, and all the funding agencies as well that have always funded all of my work. I'm really grateful to them. And finally, my old group at UL. Thanks everyone, and thanks again for giving me the invite to talk. And if you have any questions, I'm more than happy to go through them.
Thank you very much, Shelley, for very interesting work. I actually like the story behind the science as well, so thank you for that. We have plenty of time for questions, so keep sending your questions. I will start with a question from Veronica. So she says, hello, Shelley, super cool. Have you considered measuring AFM topography in SEM chambers simultaneously while using E-beam to change the surface properties?
Yeah, I would absolutely love to do that. That's a really good idea. That would be great to do. Yeah. I mean, why not? I think there's just so many things to do else in the SEM as well. And I think we always jump to that atomic scale when we can learn so much within the SEM. We can also get much more things inside there. So environmental conditions, we can get more lasers in, because there's also some studies showing that by shining light as well on some of these ferroelectrics, these topologies, that themselves, and that you're able to move them as well, and also change the properties of these as well. So nope, that would be great to do.
Good. So my other question is about the quantification. So you mentioned about that, like what your tips for the analysis of these images, what tools you found useful, or features that you found most informative?
So for the analysis, I think it's really important that we kind of all are moving towards open access. That's my kind of policy on it. Because the reason why is you don't have the black box. It also means that it doesn't matter how much funding you have, everyone has access to the same stuff. I like as well that on, let's say in that case, GitHub, that you're able to talk to people, or HyperSpy. You can say, I actually don't understand this, or maybe that code, it's not working for me. But everyone's so helpful because it's a really nice community. So for me, get used to Python. It's not as scary as it sounds, like, especially if you're not a big coding person. It's not awful. And I think it picks something that is really important to your research. So, and I think you'll learn it easier that way, but always reach out. I think that would be my advice. Just reach out, because there's no stupid question. I'm not a coder, but I find it really handy to be able to go on to, let's say, HyperSpy and ask different people that have developed that code. It would be the same thing as if you wrote a paper and then no one ever, read it.
Yeah.
Coders are so happy when you ask them questions. So yeah.
Good. So another question you mentioned in your talk, about how to make the domain walls more stable. Can you tell us a bit more about that?
Yeah, so what I eventually figured out was that there's a relationship between the polarization direction of the domain and then the incoming STEM probe. So the incoming STEM probe has its own electric fields. I guess it has its own polarization. So in my case, if I brought the STEM probe in parallel to the direction of the polarization, after several scans, and if I kept the dose rate the same, I kept all those conditions the same, it would stabilize. Then it depended also for me what type of domain wall it was. So if it was a conducting wall, it always wanted to be in that highly charged state. And then if it was an insulating wall, it would like to turn into the other direction. So that's how I would stabilize it. Another option that I started using was to use the in situ biasing holder to apply a bias that was higher bias than the STEM probe itself. So let's say you have a type of domain wall that doesn't like to stay stable, then you can overcome that by using an in situ biasing holder in that case, and then switching that off and comparing those two. But yeah.
Okay, very, very useful tips. Thank you. So there is no more questions. So I would like to finish by thanking you for joining us and sharing your science and the story behind.
Thanks so much again.
So I think I will hand now to Liam. Is that right?
Yeah, I can take it and immediately pass the baton on to Emma and Rhiannon. So yeah, I'd just like to reiterate everything that Hiba said. Thank you to the speakers for that first session. There was some really mixed bag of science there, and it was all really great. Covered a lot of diverse themes, and I think that really reflects all of us as well in the field. So yeah, I'll shut up now, and I will hand over to Emma and Rhiannon to introduce the panel, how that's going to work, and yeah, do you want to take it away?
Yeah, thanks very much, Liam. And thanks for so far all the great talks. It's been really diverse and everyone's really enthusiastic about their science. So it's great to see and have such a good crowd here as well. So I'm going to kick off the panel discussion. So we have a great selection of excellent panelists from lots of different scientific backgrounds that we're going to have a chance to talk to today. So if you have any questions, feel free to either pop them into the chat, or if you want to ask questions anonymously, you can do that via the Padlet, which Rhiannon has just shared in the chat there. So first of all, we're just going to meet the panelists. So I'll just ask the panelists to turn on their cameras, and I'll just run through the list I have here. And if I could just ask you to introduce yourselves and what is your current job. So first we'll go to Deirdre.
Hello.
Hi, Deirdre.
Hi, you can hear me. Hi everyone. My name is Deirdre, or Dee Kavanagh, actually, people know me by. And yeah, first of all, to say it's really nice to be here to share experiences and to answer any questions. And thank you for the invitation to take part. So I'm recently started a new position as a facility manager in the biochemistry department at Oxford University. Prior to that, I was working as an imaging specialist at the University of Birmingham. And before that, actually, I did a postdoc for 5 years. So I haven't had like the most straightforward route into kind of finding, I guess, what I wanted to do in my career. And prior to being a postdoc, I worked as I did a PhD in engineering and physical sciences. And I've just realized actually that it's 10 years since I passed my Viva. So I can't believe actually a whole decade has gone just like that so fast. So yeah, that's me. That's my introduction for now.
That's great. Thanks so much, Deirdre. So one of the topics we'll be covering is career pathways. So there will be an opportunity to ask all of our panelists more details into how they got to where they are today and all of those kind of questions. Thanks. So next we'll get on to Daniel.
Hi everyone. Can you hear me okay?
Yes, thanks, Daniel.
Okay. Yeah, hi, I'm Daniel. I'm an associate editor at a journal in the Nature research portfolio called Communications Physics. We publish basically across the whole discipline of physical sciences. I've been there for about 2 years. And before that, I was in academia, so I did experimental physics.
Thank you, Daniel. Next, we'll go on to Lindsey, please.
Can you hear me okay?
Yes, we can hear you. Thanks.
Sorry, I'm not overly familiar with Zoom, so
no, it's perfect. We can see you fine, hear you.
Thanks. My name's Lindsey Burton. I am Global Sales Manager at Call LED. I've spent most of my career in photonics related applications. A significant part of that has been in microscopy. And early in my career, I was in research, but yeah, moved into industry. So I'm flying the industry flag. Thank you.
Thank you, Lindsey. And next, Michel.
Hello, well, I'm feeling very old. I'm a professor of cell biology at the University of Leeds. So I've had the very standard, typical research path of PhD. I think I had 3 short postdocs over a year to a couple of years. Then I had a Royal Society University Research Fellowship. Then I got a lectureship. And eventually, after many years, I am now a professor, but I do lots of other things as well. So I don't just sit in my office. I do lots of other things.
Brilliant. Thank you, Michel. And finally, we have Liza, or Lisa, sorry.
Yes, I am Lisa. Hi, I'm a science artist. So that's a bit of a weird position to have. So rather than an industry or an academia flag, I'm sort of like flying the freelance flag, which says you can be anything you want. If industry, academia doesn't work for you, you can just do what you want. So I became a science artist and I make both science and fantasy art. And it's beautiful to see how these two interlap gorgeously. Because one of the most important things of science is to actually get your work out there and have people understand you and your message, and not just understand you, but have this emotional connection. And that's where I come in, and a lot of people who work in science communication, because yeah, art is a story and a form of storytelling. And I work very close with a lot of science communicators and storytellers. So it's a very fun line of work to be in, because we're always talking about like, oh, how I want to tell the story and how do we get people excited? And there's a lot of beautiful and fun things involved. So my background is in science. I did a PhD in microbiology and I worked as a postdoc in the United Kingdom for a while. And then I decided, you know what, I'll do what I want. I'll follow my dreams. Like a dumbass, but it's been good.
That's very cool. Thank you so much for introducing yourself, and thanks to all the panelists so far. So to all of our listeners out there, you can see that we've got a great selection of panelists from lots of different types of backgrounds. So feel free to pop the questions in, and Rhiannon and myself will try and get through them as much as we can. So a few weeks ago, we put out a tweet. We started a Twitter poll to see what early career researchers out there would like us to ask in this panel discussion. So we put out a few options, and the 2 topics that came back with the most votes were career pathways and what was the other one? Sorry, that's on the other page. Securing funding as an early career researcher. So we're going to start off the first half of this panel discussion talking about career pathways. Everyone has a different career, everyone has different journeys that they go on to get to where they are. They might be something that they've dreamed of since they were small, it might be a surprise. So first, I'm just going to ask the panelists to give a rundown of their career pathways, what brought them to where they were, was it where you expected to be? So we'll go to Daniel first, please.
Thanks. So I guess I would say my kind of journey to where I am now started when I did my undergraduate degree. I did a 4-year integrated master's at University of Nottingham in physics. And I got into that because I was really interested in physics, and partly to spite my A-level teacher who told me I was never good enough to be a physicist. So it's a little bit of like trying to prove myself there. And I didn't expect to do a PhD, but in the summer between my 3rd and 4th year, I did an internship at a neutron scattering facility in France, and there I really enjoyed not just like learning textbook physics, but actually doing research with people who were trying to find out something new. And I thought, okay, if I can do that, then that will be really interesting for me. So I decided to do a PhD after my 4th year, and I moved to University College London. I was based in the chemistry department, but we were all physicists in my group. So most of my group were doing STM measurements of molecular absorbase at surfaces. And I was doing ultra-fast laser spectroscopy on the same surfaces. Then after that, I moved to do a postdoc in Italy at another ultra-fast laser facility. And I was there for about a year and a half. And I don't know if anyone has worked with ultra-high vacuum equipment and ultra-fast lasers, but those are two machines that just don't like each other, and they're incredibly temperamental. And after about a year and a half of banging my head against the USB chamber, I just couldn't stand it anymore. And I realized that what I liked was the analytical side of things, the piecing together the evidence, trying to understand a coherent picture of what was going on. And what I absolutely hated was taking things apart in the lab to try and figure out how they'd broken and how I could fix them. So I decided to move out of experimental work and applied for this job as an editor at Springer Nature. And one of the things that was really cool about that is that instead of looking at a really small area of research where I was trying to know everything about that particular topic, I was now encountering research across a much broader field. So I went into handling the optics and photonics papers mainly, but we sometimes need to handle any topic that comes in. So it could be biophysics, it could be astronomy. We really could be varied on a daily basis, and that's quite cool because you're always learning something new. And it's really sort of, instead of going very deep into a topic, you go shallower but broad. So that's where I am now. I've been here for about 2 years. I don't know what I'll do next, but maybe we'll see.
Thank you, Daniel. So we might just take a question that's come up on Padlet, and then we'll continue on with the career pathway discussion. So I can see the first question here. I am interested to know how much job security has played or does play into your career choice. So Michel, maybe we'll ask you that question.
I think that's a really hard question, because actually as a PhD student and postdoc, I thought science was brilliant because it gave you the opportunity to travel. I went and worked in the US, I went and worked in different places. We've heard already about Daniel going off and working abroad. So that for me, I didn't really care so much about the job security because I could go and work where I wanted, and it was only for a short period, but it was a great experience. It's like so I think it's probably when you're sort of settling down and you've got a partner and it's difficult with two people to find jobs in the same place and all of that kind of thing, where job security starts to come in and be a bit more of an issue. So I was lucky because at that point I did get a fellowship, which in theory would have given me up to 10 years of funding, certainly 5 to begin with. And then it just and then you just get used to it. I saw one of the other questions about stress of academia, and of course the problem is that stress never goes away, because even as a professor I'm always having to get grants, and of course a lot of the time the grants are turned down, and all the time I'm worried about the staff in my lab and how they are going to have job security and whether or not I can keep them employed or so it never goes away. But I think it's partly what stage you are at and how important to you that job security is. And I think there is a lot more concern about this in universities now. Universities do have redeployment strategies. If you can't work for that PI, you could go and work for another PI in the same place. There is a lot more support for that kind of thing, but it's never going to be until you actually get that job as a lecturer, it's always going to be some sort of job insecurity, isn't there?
Yes, yes.
So,
It's very much an individual question.
I think it's a personal choice how much it bothers you. That's right. Yeah.
Yeah, definitely. Any of the other panelists like to have a go at answering that question about job security? Yeah, Lisa?
For me, job security was a massive contribution for taking this step from science to becoming a freelancer, which is a bit of a weird thing, because one of the problems with being a freelancer is that I am super dependent on actually getting jobs. And when I worked in science, I had a really lovely monthly salary, which was absolutely amazing. It's really like, I don't think I appreciated getting like monthly salary enough at the time. But at the same time, I can now, working from home and working anywhere I want, I can pick up my drawing tablet, go anywhere, and I feel like I have a lot more freedom, and I feel more secure in that sense.
Okay. Yeah. So your security wasn't so much tied into your wage, it was more security of being able to.
Well, for me, I got a lot of stress from the idea like, oh, what if I finish this postdoc and then my ultimate dream postdoc is in a different country where I then again have to meet new people and make new friends. And for me as a person, that was super stressful. So the idea that I can now have a freedom to work wherever I want is very nice.
That's your security. Thank you very much. Deirdre, you have your hand up there.
Yeah, actually, I just wanted to kind of echo what Michelle says. Like when I was younger, I didn't really mind, you know, having the contract as the postdoc. It was kind of nice to be able to have that flexibility. And I thought it would matter more as I've got older. And just for me personally, and my recent experience was that I had permanent job offers, but actually the right job came along for me just recently with really good career prospects. And it is a contract. So in the end, I ended up accepting another kind of fixed term role, which I didn't think I would. It's just because I know this is what I want to do. And basically it opens up better career options for me at a later date. So that was another factor kind of that came into play, and I didn't actually consider it beforehand.
Yeah, sometimes unexpected things come up that you might not have thought of. So if we might just continue with our career pathway kind of topic. Lindsey, would you be able to give us a brief overview of your career pathway?
Sure. Did I plan to be where I am now? No, not at all. I went off to university and did what I liked, which was biology, did my undergrad. But it was during that time that probably, with hindsight, drove where I ended up. So I had an opportunity to do a sandwich year and work in clinical labs in a hospital. And I went and spent a day in each of these labs to decide where I wanted to spend my year. And I had originally thought I'd go to the biochemistry labs, but I realised that was just a load of boxes you press buttons on. And I ended up in the pathology labs, playing on microscopes. So this was way beyond what I'd ever used at school. So I was pretty excited by this. So I spent a year in a hospital lab using microscopes, using EMs and fluorescence, even back then in the 90s. And then after my undergrad, I followed the path of everybody else. I went into research and pretty quickly realised it probably wasn't really for me. So pretty much like Daniel, I wasn't too interested in the fact of focusing down solely onto one area. And, whilst I was wondering what I was going to do with my life, because I didn't really know what other career path was available, we bought a big microscope system. We had the salesperson come in and we had the trainer come in. And that kind of opened me up to the world beyond academia. And yeah, I guess the final decision for me was the lab that I worked in flooded, and our work was annihilated with a lot of researchers. So that was back in 2000. So when we did return back to the lab and we realised we're going to have to repeat quite a considerable amount of work, I decided I was going to leave. And so I spent a bit of time talking to salespeople that came into the labs and asking them what their experience was like, and I shadowed them. And I took the plunge to leave, and I was got a job with a company called Hamamatsu, and they deal with cameras or photo multiplier tubes. So I got very much involved in light detection systems. And then I spent 5 years working for them. They're a really good company in terms of opportunity to learn and develop. And then I went on to work for Andor Technology. So another camera-based company in microscopy, but they also sell spectrographs for spectroscopy. So I got involved in other types of photon detection, and but still went back to microscopy. I started selling spinning disk confocal systems, and I did that for several years before actually I took a bit of a career break and went backpacking, which is something I always wanted to do. And then I went back to Andor a few years later and did a lot of spectroscopy and astronomy stuff then, so linked to microscopy, but in reverse. And yeah, after another several years with them, I ended up joining CoolEd. And my interest in joining CoolEd was a different type of sales opportunity. So now I was selling the light source on the other side of the microscope, and also dealing predominantly with other salespeople and other reps. So that's been really good. So yeah, I didn't plan to be here, but I've had a really interesting career. I like the fact it opened up the possibility to talk to other researchers about completely different sciences that I'd never thought I'd be interested in or know about. And the types of people that I get to meet are amazing. You know, I've had conversations with Nobel Prize winners. 1 minute you're talking to astronomers, the next you're talking to a biologist. And for me, it's the variety that I've loved about the job that I do. Yeah.
That's great. Thank you so much, Lindsey. So we have a question here on Padlet that's had quite a couple of likes. So it just says, hi, I'd be really interested to know how Lisa found the jump from academia to entrepreneurship. How scared on a scale of 1 to 10 were you before making the jump? And have any other panelists considered starting their own business? So Lisa, we'll start with you first.
Thank you. That's a very fun question. So considering how scared I was, it's a bit like, I'm very lucky because both my parents are freelance journalists. So I kind of knew the freelance life in a way already. So I've seen how they live, how they work and get their jobs. So in a way, working freelance for me felt more like what a job is supposed to look like than academia. But I was in no way prepared. And I found that universities are actually really good helping you prepare for your own business. Most universities have business coaches. If you're a PhD student and you're interested in starting your own business, usually your university is like really prepared to help you out. And they love it when PhD students start their own business. So whatever you're interested in doing, have a look at that. Because when I was a postdoc at the Newcastle University and I decided to leave and become a freelancer, they were also super supportive. And there's a lot more help than we often realize. And the other thing that I found intimidating, but also really important, is really having to learn my rights. In my case, that's as an artist, I have copyright. And I thought I would be able to do art, but I had to become an expert on copyright law. It's really complicated, but also really fun. And another really important thing, if you become an entrepreneur, there's only 2 things in life I'm scared of: the inevitable heat death of the universe and taxes. Taxes. I had to do my first tax return and I'm still sweating, but there's also people who can help you with this. So in the end, like, it was scary, especially losing the safety net of exactly this monthly salary. But at the same time, for me, nothing beats being able to do what I love most, which is making art. And it's really cool that I get to make people happy with this, because the best conversations I have are with scientists who have this vision for either like a cover image or a model they want in their research. And they say, I think it needs to look like this. And when you get to the moment where you have their vision on paper and are really happy, that's really what I live for. But yeah, if anyone's thinking of entrepreneurship as well, it is scary, it is weird, but embrace all the help there is, because usually, as I said, academia, it doesn't really prepare you, but they really want to help you. So just take the business coaches.
Yeah, the help is there. You just need to find it then, all right. Okay, so Daniel, you still have your hand up there?
I actually wanted to ask Lisa a question about how that transition happened. Did you take the plunge all at once, or did you slowly move away from monthly salary, you know, to increasingly spending more time freelancing?
Well, this is one of those things where the pandemic definitely played a role. When I was like not able to go to the lab, I had to do something with my time. And for me, that meant that I suddenly started creating so much more. And I found that if I can do anything I want with my time, for example, when I'm furloughed, apparently I make art. So probably that's something I need to do more than research, if that's apparently something I naturally lean towards. And yeah, I found that there's no really a way to do science part-time. I think it's technically possible, but emotionally for me, I feel like if you're in science, it's a lot and it consumes you. So yeah, I actually had a conversation with my professor at the time. My professor was Jeff Arrington. He was super supportive because I told him that my ambition eventually was to become a freelance science artist. And he basically just asked me, so when are you starting? And I hadn't really realized that I could really just quit my job and do it. So yeah, that was really amazing to just have someone say, "Oh, do it then." And it's like, yeah, watch me, I'll do it. But yeah, that was just go. I had a few jobs before I started, so I kind of like got into it, and you kind of need that renown. And for me, what really helped was a Kickstarter. So perhaps people here saw that I did a Kickstarter half a year ago for science enamel pins at the time, because one of my friends said, "Oh no, it would be cute, science enamel pins." And I said, yeah, let's do this. So I kickstarted like a set of 5 science enamel pins, and it was widely successful, which was crazy. And that really gave me the feeling of, you know what, apparently people want what I do, so let's do it.
Something that like strikes me with a lot of the stories we're hearing is that element of luck or even bad fortune with labs flooding or a pandemic that just sets you off, motivates you to go in another path. But it seems like deciding what to do at those moments comes with like an element of self-knowledge. So understanding yourself, what your values are, what your skill is that you can do maybe better than other people around you, and trying to leverage that to go forward.
It's a very beautiful way to say it, really. It's also a way of turning your bad fortune into good fortune.
Yeah. So I'm just going to go to the Padlet questions here. Let's see. Sorry, hang on 1 second. I really enjoy working in science, but I am not sure academia is for me. How easy was it to move to industry? Would I then be able to move back into academia after? So I guess I'll direct that at Lindsey.
I think a move into industry is relatively easy. I think it's about identifying what job you want to do in industry as well as. So, you know, for me, I went into sales primarily because it was what I was, I suppose, exposed to. I had a very, you know, with my career now, I know there's quite a lot of jobs that exist in industry, you know, be it an application specialist, you know, where you support customers with their particular experiments with the equipment manufactured, or you could be a product manager, you could work in R&D. There's a whole host of jobs with inside industry that, you know, I'm not necessarily an expert on, but I think taking time to investigate those a little bit more. Most of the sales reps in our industry especially are open to chatting. They're lovely guys if you want to know more. And I think that will always put you in a good position when you want to apply for a job in industry, to show that you've made an interest, that you really understand what the job is likely to entail. You know, we do occasionally get people that have an interest but really haven't grasped an understanding of really what the job is likely to entail. And I think if you can demonstrate, look into it, that will put you in a good position. I don't know about going back, I haven't tried.
So. Michelle, you have your hand up there?
Yeah, so I can just comment on that because you know, industry means a lot of different things, and of course, you know, a lot of if you're in the life sciences, and I think actually this is true for physical sciences as well, where people will go and work in industry, but then go back into academia. So I'm pretty sure your president of the RMS, Grace Burke, has gone to work in industry and is now back in academia, for example. And I can think of, you know, lots of examples where people have gone to do perhaps more of a research-based job in industry, but then have made the move back into academia at some point for whatever reason of their own. So I think it's absolutely possible. It really depends, you know. It's like the choice of whether you want to stay in academia and you know, try and get to that point where you have a bit more job security, or whether. So a lot of my postdocs have gone into industry because of the job security, and they see that as having more job security. But you know, maybe at some point they'll change their mind and come back again. Who knows?
Grace, are you? I don't know. I did see your name, Grace Burke. Would you like to chime in and give us a few words on your experience?
Oh, okay. Anyway, hi all. It's been a really interesting symposium and also discussion. Yes, I started out, I did my PhD at Imperial, and 2 weeks after my viva, got married, and my husband and I took positions in the States because there were a lot of R&D laboratories. So that was exciting, challenging, because it meant moving into some new areas. But the common thread was that electron microscopy and, well, in my case, metallurgy, was something that turns out was a bit on the unique side in terms of the techniques that we use and the ability to study the structure of materials. So I spent some time in the steel industry, which then proceeded to go down the hill. So I have the distinguished honor of being the last person to ever use the 1,000,000-volt microscope at the US Steel Research Laboratory. But then moving into Westinghouse and going into nuclear materials board, nuclear power systems, I found it to be really challenging, very exciting. And then moving into academia was a culture change for me, because I was used to the UK system when I was a PhD student. So things have changed quite a lot in terms of academia in the UK. But I think it brings with you an extra dimension in terms of the relevance of what you're teaching and the importance of the techniques and understanding things. I think it's a good mix. So it just depends what people's interests are. If you want to be involved with a lot of research, industry does have that opportunity for you. I don't know. It's a mix. It depends what you want to do. You don't get to interact with students there, so that's the benefit of academia.
That's great. Thank you very much, Grace, for having a chat with us and joining in the discussion. I suppose there's always, as an early career person myself, you always think of a trajectory of an academic career as the straight road. And if you go off that road, there's no coming back to it, you know, that kind of way. But that's not true, apparently.
No, and sometimes that, how should we say, meandering path gives you a very unique perspective and also unique skills that will come into play later on. Things that you may think are a bit out in left field will end up becoming very important. So
that's great. Thank you so much, Grace. So I'll just ask, so back to the career pathway, just very quickly, I'm kind of getting aware of the time. Deirdre, would you be able to give us a quick overview of your career pathway?
Hi, yeah, sure, definitely. Actually, when I was doing my PhD, I thought I wanted to do the whole academic thing, actually, but when I was doing my postdocs, that's when I became less sure. Because at that time when I was a postdoc, the things that I were enjoying the most were really kind of outside of research. So like at that time, we did a lot of public outreach and I was very involved with this. also I was very involved with the Postgrad Society, and also we had an imaging facility that was newly established at the time. So I worked firsthand with the manager at that time, and also met the researchers that were coming in to use the facility, and that kind of really had a direct impact on what I wanted to do for my career and really shaped it. And you could say that's kind of how I've ended up working in facilities. So yeah, I mean, it was things that I got involved with outside of research that really, I think, ended up shaping what I wanted to do in my career.
Yeah. So there's a question in the Padlet that maybe you might be able to answer. So it says, I'm really curious to learn more about working in running facility. What is the bulk of your work? Are you mainly building running microscopes or helping other scientists run design experiments? And what motivated you to work in a facility?
Yeah, that's a really good question, actually. And I think, you know, what I love about it is there is so much variety, you know, so you do a lot of hands-on stuff at microscopes. You know, you're doing a lot of teaching and training, you're interacting directly with people, you know, and that's, I love all that, basically engaging with people and teaching them microscopy. You're also like fixing microscopes, doing maintenance, doing quality control. You work with companies, all the microscopy companies, the vendors, you know, the engineers come in, you know, to service the microscopes. You're responsible for health and safety, finances now, you know, because the facilities often have to recover their budget. So that will pay for the staffing and basically maintenance on the microscope. So yeah, it's a real mix of different things. And actually, you know, you have to have skills that necessarily you don't pick up as a PhD student or as a postdoc, and especially management skills. And I actually had to source some management courses and go on them to help me. So yeah, that you know, these are kind of what you need, you know, the finance stuff and the management. I went on some courses last year, leadership and management courses that were really useful. And I think they just really made a difference to get myself in the headset of like managing a big facility. Yeah. And what was the last part of the question, I think?
The last part was, what motivated you to work in a facility?
Oh, that was work, you know, like I said earlier, just actually seeing how a facility is run, and, you know, just seeing the impacts that it makes to researchers when they come in. And yeah, the first-hand experience. I think I was quite lucky at being in the right place at the right time, that I was exposed to that. Yeah.
That's great. Thank you so much. So I'm just going to hand over to Rhiannon now, and she's going to take the second half of our discussion. Rhiannon, are you there?
Brilliant. Thanks very much, Emma. And thanks to everyone that's brought in questions so far. We've had lots of interesting things on early careers. And we've also touched a little bit about careers feeling unstable. And that kind of moves us into funding and how, as early career researchers, you can move towards getting these funding grants. And we kind of have a look at the Padlet. There's a more specific questions going on, but I guess at the moment, the question I'd like to ask the panel is, how would you recommend as an early career researcher going about getting funding so that you could move into a PI or fellowship role? Michel, if you want to kick us off, kind of how your process occurred.
Yeah, I can actually speak about this from two sides, because I actually have ended up sitting on quite a lot of, so I've done MRC early career development fellowships, and I do UKRI Future Leaders fellowships at the moment. And the kind of career, I mean, the thing is, like everything in science, is you've got to have your own little niche, really. You've got to have something that A, you find interesting, and B, everybody else does as well. So it has to be something that you can sell to somebody else, and you've got the right career development experience and connections to pursue what you want to do. A bit like becoming a freelance artist, actually. You know, you've got to know what you want to do, why you want to do it, who's going to support you, and how you're going to make it a success. So the career path after that is, you know, I mean, I think, so for me, I'd done a number of different techniques. The final postdoc I'd done had started to exploit the use of molecular biology to introduce mutations so I could look at disease processes, but it was all in fruit flies and I wanted to do it in a mammalian system. So I was kind of interested in developing that. And then at the same time, somebody that was, in fact, my first postdoc supervisor was just chatting to me and saying, why don't you apply for this fellowship? You could come back to King's where I was for my first postdoc, and you know, it would be a great place to do this. So it is that sort of going and talking to people and networking as well that kind of helps you decide what you want to do and how you want to do it. And when you actually see people's coming in applying for fellowships, you know, it is a very mixed bag, but it's typically building on what you've done as a postdoc, finalizing or working towards that sort of really key question you want to address, the kinds of techniques that you've built up as you've been developing as a postdoc, having that question or set of questions that you really want to answer that perhaps nobody else is doing but you see as really important. And getting support from other, you know, going and talking to people, getting as much support from other people as you can to put it together. The fellowships are always about person, project, place. So it's you as a person, how you've developed your career so far, where you're going to do the research, how it's going to support you, and what the project's about, and you know why it's important. So those are the kind of things to think about, and it can take a while. It really does take a while to come up with the strategy that you're going to use if you want to apply for a fellowship. So it's a good, I would say, 18 months before you possibly even put that application in, and be prepared to be disappointed. You know, I didn't get my first fellowship. I applied to the Royal Society. I didn't get it the first year, so I had to apply again the second year, and then I was lucky. So, you know, and you see this, people apply for lots of fellowships. If they're really determined, you know, you just keep applying. Is that helpful?
Yeah, that was really nice and succinct. Thanks, Michelle. I guess the key message from here is, you know, demonstrate your passion and keep persevering if that's the career path you want to take. Do any of our other panelists want to add anything to that in terms of that side of funding? Otherwise, I wanted to look more into the technicals. We've got an interesting question here about securing funding as an early career or like as technician or facility staff. So I wondered, Deirdre, if you'd had any experience with how that works if you're a facilities manager.
Actually, yeah, so as a facilities manager or working in an imaging facility, you're usually on an academic related contract or similar kind of to a postdoc contract, which can be good and not so good for different reasons. So where I've always worked, I've had actually no problem in you know, applying for applications and, you know, being named on grants. So I've always been supported in that way. I've not come across any like problems with that. Where I've been successful, and I think this applies to anyone that's just starting off, is and people would probably agree, is just the small project grants or the internal project grants. Basically, you know, in terms of like what I would apply for typically is like something to hold a workshop or something like that, or, you know, some kind of course. That's mainly where I've applied for funding rather than your research-based grants. Yeah. So, you know, in terms of like, I've applied to the European Microscopy Society, the Competent Biologists, things like that, to just basically collect small pots of money for holding courses and workshops. Yeah. Yeah, so that would be my view, really.
Yeah, exactly. So thanks very much. Just so it kind of shows we've obviously got our big fellowship grants, but alongside those, there are also lots of other smaller pots of money that if you're already in a postdoc, already in a facilities management or technician space, you're able to look into those.
Yeah, exactly. And the other type of grants would be infrastructure grants that you would have to being applied to in these type of roles, you know, so you can always have the equipment and keep that basically up to date. Yeah, to remain competitive.
Perfect.
Yeah.
And I guess that moves us on to the other sort of funding, which is considering, we've got a question here about collaborating with industry and how easy or how positively it's viewed, I suppose, from an industry side. to be able to collaborate as an early career researcher and get funding. So I don't know if Lindsey, you could answer that from an industry side at all.
I'll certainly try, but we obviously do get approached as an industry to support PhD students, etc. In fact, Liam, who's on here, might be able to comment better. But it's largely taken over by another section of the business. It's usually part of our R&D team, for example, that are wanting to work with the universities to do it. So yeah, we certainly do it. There are some grants available that kind of benefit both parties. I don't know too much about them, I'm afraid. But it's something, certainly as a small company, we do value those links that we have with universities in terms of developing technologies and applications. But unfortunately, from my point of view, I don't have too much direct involvement, so it's quite difficult for me to give too much advice.
That's absolutely fine. I don't know if I know Grace, is she still available? Said she would be happy to answer any questions in working industry versus academia. I don't know if you'd be able to give any insight into this.
Well, I mean, one of the benefits of having, most of my career has been spent in industry, in R&D environment. And basically what it tunes you into are the things that are most relevant and a very targeted approach in terms of going after solutions or what aspects of the work is required. So basically it gives you excellent links with industries. Even my activities in the States, I'd already been working closely with people at Rolls-Royce and Serco, AMEC, I guess Jacobs now. So basically it tunes you in right away. And the thing that I always keep in mind is that there are issues or problems to solve, and it's not that industry is looking to fund you to do something you want. You need to be doing something relevant to industry, and that will serve you very, very well in terms of developing successful and growing relationships. And that is materialized in terms of funding. So I hope that answers questions. I'm happy to talk to any of you offline if you'd like to chat a bit more about it.
Perfect. Thank you very much, Grace. I noticed, Michelle, you had your hand up. Or has it all been covered?
Well, I think there are a couple of things that have come up, but as I've been typing in the chat, I've probably forgotten about it. But I think the industry thing, certainly that's actually quite a good thing if you're applying for a fellowship and you have got some industry link up in whatever shape or form, as long as it's justified and fits with the project. So we see quite a few of those where that person might spend some time in industry or might have them as a collaborator working on a specific problem that industry is really interested in. So that, that's a good thing because it really kind of shows the practical applications of what you're doing. I think there was one question I've been trying to answer in the chat about how long before you actually apply for a fellowship, and I think that's just how long is a piece of string, because it used to be you could only apply up to about 10 years post PhD, but now there's no limit. You can apply at any time. So even if you're 12 or 13 or 15 years past postdoc, if it's the right time to apply, you can give it a go. Or you can apply. Sometimes we see applications and people have only been 2 years out of their postdoc, so it just depends whether it's the right time, you've got the right project, the right place, the right support, and so on. I think those are the couple of things I just thought I would comment on, but I've been trying to answer them in the chat as well.
Yeah, that's perfect. Thank you, Michelle. And Daniel, you've got your hand up. Would you like to add something?
Yeah, I just wanted to add a little bit about the advice I picked up. I tried to crowdsource this question, so I put it out on Twitter, and I promised that I would share the responses with people. So these are tips that were given by current PIs who I've come across, through my interactions with the journal. So hopefully they'll be useful for some people here as well. So one of the main things that came up was the importance of having a PI or a supervisor who actively support you building your own independent career. They need to be not only tolerant of you spending time on this, but actually support you, encourage you, give you feedback. And I think maybe if you are at the level where you're going to start spending 18 months on a proposal that you want to submit, then it's probably important to be very clear with the supervisor from the beginning that this is what you want to do, and are they going to support you, and really get that relationship dynamic right from the start. Then the other thing that was suggested was to try and obtain examples from your friends and your colleagues who have had successfully funded grants. Really try and look at the structure they've used, the way that they've phrased it, the way that they talked about their research. And then when it comes to writing your own, to try and get advice from people at all career levels. I don't know, I haven't done this personally, but what I understand is that when you're on these grant panels, you don't have much time to assess each proposal. So you want to give it really clear and succinct, and you want undergraduate students to understand it, because the people assessing it are not necessarily experts on the topic that you're researching, that specific niche of it exactly. And even share it with your friends and family. See if they can understand it, then there's a very good chance that the proposal panel will quickly get why you want this position and why you think it's important. And then the last bit, if I can just
go for it, last one.
Yeah, was just to focus on getting what is necessary. So forget about the nice to haves, forget about if you just spent another month on this, you could get a bit of extra money for X, Y, Z. Just get what you need, get the results and move on, because you really need to be pragmatic and just focus on achieving, essentially.
Brilliant. I think that is a pretty good summary from Daniel there about some of the pitfalls. I'm afraid we're out of time, but if you do have any more questions, we will be putting an email in the chat and you can send them through and we'll make sure they'll get to our panelists if you'd be happy to answer any further questions that we have. But I'd like to thank you all very much for your time, and I'll hand over to Liam just before we finish. So thank you again to our panelists.
Yeah, thank you very much, everyone. That was a really great panel discussion. I think we had so many unanswered questions in the Padlet that are still there, so we'll try our best to get them sent around to everybody. And yeah, thank you for everybody's engagement there. That was really good. So now we'll head off to a break. Let me just put up a holding slide so that we know what's next. And yeah, I'll welcome everybody back here at past. So please go make yourself a tea, a coffee, grab a biscuit. Don't eat your hotel chocolates for Wednesday. I'm looking at you, Grace. Yeah, I'll see you back here at past. Okay, catch you later. Good afternoon, everybody. Hopefully everybody is kind of siphoning back into their seats. We're due to start the next session in about a minute or so. So I'll just give a brief overview before handing over to our session chair. So we have some contributed early career talks, followed by our Early Career Award lecture from Kevin. And then I please urge you all to stick around for the RMS Early Career section, AGM. We'll be able to kind of update you on the activities of the committee over the past year. And we'll also be announcing the winner of the prize for the best oral presentation from our contributed speakers today. So please stick around for that. And without further delay, I'll hand over to our session chair, who's Dr. Becky Salib from the University of Edinburgh. And Becky, if you want to take it away for the next session, that would be fab. Thank you. And I'll stop sharing.
Thanks, Liam. So first up in this session is a talk by Sfi Islam, one of our contributed talks. Asifa is a PhD student at the School of Biological and Chemical Sciences at Queen Mary, University of London. She'll be sharing with us her work investigating human genomic variations in aspirin and their impact on chromosome segregation using single-cell live microscopy. So Asifa, if you could share your screen now and take it away.
Yeah, hi. Okay, so can you see my screen now?
Yeah, perfect. If you could just put it into presenter and presentation mode.
Hey everyone, can you see my screen?
Perfect, yeah.
Yeah. Right, so hi everyone, my name is Asifa and I'm actually from a medical background, but right now I'm doing my PhD in Professor Vijit Rabham's lab in Queen Mary, University of London, where I use microscopy to study variations in diseases such as cancer and developmental disorders. So we have mitosis lab, and here I'm showing you a cell in mitosis. So what happens in mitosis is that our chromosomes here in blue are captured by these duplex structures with the help of a multi-protein complex called the kinetochore. Now kinetochores are a group of group of proteins, which is like more than 100. And genomic variations in kinetochore genes can cause cancer, but they can also cause microcephaly, wherein children are born with small head and have intellectual disabilities and developmental disorders. Genomic variations in kinetochore genes can also cause mosaic variegated aneuploidy, where children have microcephaly, but they are also at a high risk of early onset cancer. Now, there is a higher incidence of microcephaly in South Asians due to prevalence of consanguineous marriages. And that's why I've been working with Genes and Health, which is a population genetic study of eukaryotes of South Asian ancestry to identify genomic variants in kinetochore proteins and to study their impact. At the scope of my PhD project, I have focused on a protein called aspirin, which is already implicated in microcephaly in this paper that was published in 2019. And when I looked at Genes and Health database, I found 3 loss of function variants of aspirin, 2 in the N-terminus and 1 in the C-terminus. In this talk, I'm only going to talk to you about this C-terminal variant. There are only 2 individuals with this variation, and both of them are heterozygous. And I didn't find this variation anywhere else other than just East London. So what does this variation do? Basically, there's a generation of a stop codon, and which means that this very C-terminus part of the aspirin will not be expressed. And why is the C-terminal of aspirin important? It's important because it's required for the localization of this 4-unit complex at the kinetochore. And also, we have shown in our 2019 paper that the C-terminus of aspirin procures PP1 phosphatase at the kinetochores. And this is important for the stability of kinetochore microtubule attachments. And only when stable kinetochore microtubule attachments are formed, then spindle assembly checkpoint proteins, which mediate kinetochore microtubule attachments, can leave the kinetochore, and the cell can proceed into anaphase, and 2 daughter cells can form. Now, this variant that I identified in the Genes and Health database, it lacks this domain, which has the PP1 phosphatase docking region, and also this region is required for the localization of this complex. So my first question was whether this variant can localize normally. And to ask that, what I did was I seeded mammalian cells, HeLa cells, and I treated them with aspirin siRNA to deplete the cells of endogenous aspirin. So endogenous aspirin runs as a doublet on a Western blot. And you can see here, when the cells were treated with the siRNA, the doublet is lost. Next, I transfected these cells with the plasma encoding these variants. And you can see here, this is a wild type and this is the variant. And again, you can't see the endogenous aspirin bands. Next, I arrested these cells in metaphase by a drug called MG132. This is because we know from our previous studies that aspirin levels at the kinetochores are highest in metaphase. Finally, I fixed and immunostained these cells. And here are the images. So here DNA is in blue and the centromeres are in red. Centromeres are these regions on the chromosomes where the kinetochore will assemble. And you can see here estrogen in green is decorating the microtubules, but also co-localizing with the centromeres. So giving off this yellow color, which means that estrogen is localizing at the kinetochores. And now if you have a look at the variant is present on the microtubules, but it's not present at the kinetochores. So once this was established that the variant fails to localize at the kinetochores, the next question was obviously, would that have a functional impact? And to ask that, I looked at metabolic progression, which related this HeLa radio conditionally expressing cell line to white aggregates and stuff, which can impair the localization of proteins. And once I had this cell line, I see the cells use double thymidine clock to synchronize the cells and use tetracycline to induce the expression of these estrogen in these cells. And I also use cell DNA, which is a diet by chromosomes, and then finally use live cell imaging. So here I'm showing you the time-lapse images from the videos that I generated, and you can see this is the wild type. First, the nuclear envelope breaks down and the chromosomes congress and align at metaphase here at 18 minutes in the cell, and then finally segregate around 42 minutes in the cell. Now, if you have a look at the variant, again, first the nuclear envelope breaks down, chromosomes congress and align. This is happening at 96 minutes in this cell, and segregation is happening around 168 minutes in this cell. And if you have a look at the cumulative frequency graph, you can clearly see that there is a delayed anaphase onset in the variant expressing cells. Also, if you look at these three images here, you can see there are unaligned chromosomes here, and also there is this lagging chromosome in this cell. And when I quantified cells with lagging chromosomes in anaphase, what I found was that less than 20% wild-type expressing cells had lagging chromosomes in anaphase, whereas the numbers were really high in the Q1 and 1-2 expressing cells. So the variant fails to localize at the kinetochores, and there are unaligned chromosomes or lagging chromosomes. So the next question was whether the expression of this variant is having an impact on the microtubule pulling forces, because we know estrogen is required for the stabilization of kinetochore microtubule attachments. So what I did was I seeded HeLa cells again and transfected them with 2 plasmids, which I'm going to show you later, and then again, arrested the cells with MM2022, and then from that high resolution live cell microscopy. And here I'm showing you this is the wild-type expressing cell. You can see estrogen here on the microtubules and also on the kinetochores here. And this is the Q1 variant. You can see it's on the microtubules, but not at the kinetochores. So what I did is I to see whether there is an effect of the chromosome pulling forcing. I measured the distances between these centromeres because when the microtubules are going to apply pulling forces, they are going to stretch the centromeres. And here I'm going to show you this quantification. You can see here in the wild-type cell, the distances range from 0.3 to 1.7 micrometer in the cell, and in the Q1 variant, the distances were significantly reduced. So to conclude, I have shown you here 3 loss of function variants of a protein estrogen identified in the Genes and Health study. And I've also shown you that this variant is a true loss of function variant because it fails to localize at the kinetochores, and also because the cells that are expressing this variant fail to generate chromosome pulling forces and have prolonged mitosis, chromosome compression, and segregation errors. This may explain why this variant is rare and only seen as heterozygous, because you can imagine this being expressed as homozygous, and you will have all these defects, and it's unlikely that the person would be normal. And these variants were identified from individuals who are normal, that is, they don't have any disease. But of course, we have to do feature work where we have to analyze these impact of these variants by mRNA and protein expression studies from the cells from these human volunteers, because sometimes there is a variation, but it may not be present at the protein level. Right. So finally, I want to acknowledge my supervisor and my lab members and also my collaborators. Huge thanks to Emily who trained me, and also my master's students whom I trained and who contributed to this work. Thank you.
Thanks, Asifa. That was a really interesting talk. You can stop sharing your screen so we can see you. If anyone has questions, please feel free to put them in the chat. Liam's asking a question. Are there any drugs identified that can reverse the effects of Q1012 in vitro?
Not that I know, because this is the first time that variation is identified. So no.
So lots of scope for future translational work. I was also interested, you specifically chose the C-terminal variation. What drew you to that as opposed to the N-terminal ones?
So I have worked on the N-terminal variation, but I just didn't share it. But the C-terminal variant is also because we have been working on this C-terminal part of question in the lab, and we have a lot of information on why it's important. We know that it's important for the localization. We also that it's important for the function of estrogen antikinetics.
Makes sense. And as another side of things, I mean, we focus a lot on the biology application. How have you found the imaging? Doing live cell imaging is very difficult, and I imagine to our audience, it would be interesting to hear the challenges you've had in this particular application.
Yeah, because because of phototoxicity, so you have to so for my live cell imaging, because it was 10 hours long, so I had to make sure that it's very low res, which means that the fluorescence isn't always great. And so I had a bit of struggle with making those cell lines where I could see good expression, which I could also see in live cell movies. And also because sometimes, the cells just die. So you have to make sure that it's very low res. Also you have to be very careful with setting up the experiment and make sure you have a lot of cells. Yeah.
Sure. Has it impacted your quantifications at all, trying to manage all of it?
No, I think it's more I spent a lot of time setting up the experiment, and once I had the videos, that was okay, this is not easy part. Analysis is the easy part. It's the setting up is the hard part.
Well, it's a really impressive story. Thank you so much, Asifa. There's no more questions in the chat, so I'm going to move on to our next speaker.
Thank you.
Our next speaker today is Sebastian Agoyo. He will be joining us from the Pontifical Catholic University of Chile. He's presenting his work that uses atomic force microscopy to characterize early adhesion of oral streptococci to cross-linked type 1 collagen. And I can see you're already there, Sebastian, and sharing your screen. So please go ahead, pop it into presentation and take it away.
Thank you very much. Can you hear me?
Yes, can hear you.
Yes. Thank you. Well, thank you very much for the presentation and for this opportunity to talk to you guys a bit about what we've been doing in the lab in the last couple of years of using atomic force microscopy to study how oral bacteria attach to collagen surfaces within the context of dental caries and dental diseases, especially since I'm a dentist by formation, but then I've gone into PhD in microscopy a couple of years ago. So it's been a very interesting journey. So why are biofilms important for us as dentists? Well, these biofilms, which are bacterial communities that attach to surfaces, including teeth and tooth surfaces, actually are involved with certain diseases. They can secrete this EPS, polysaccharide matrix, that allows them to protect themselves from removal, for example, with your toothbrush, and it also gives them antibiotic resistance. So it's involved in several diseases that are really important. Now, within this process of biofilm formation, it's always important to remember that the attachment of bacteria to surfaces is a crucial initial step. If we are able to stop this process, we will maybe find new ways to fight these diseases. Now, in dentistry, these biofilms are associated to several diseases, but one of them is dental caries. Dental caries is this disease that is associated to the destruction of the tooth, the destruction of the mineral part of the tooth. It's associated with tooth pain, toothaches. If you ever had a really bad toothache and needed a filling, you've probably suffered from dental caries. And it is estimated that about a third of the population, at least, has this disease. So the impact on the medical healthcare, the economical part, and also even psychological component in some patients is very, very big. So therefore, we're trying to find and understand a bacterial attachment within the context of bioinformation to hopefully find new ways to prevent or treat dental caries in the population, and specifically in the elderly population. So basically what we know about the biofilm associated with dental caries is that the disease starts when there's an overgrowth of a specific bacterial strain called Streptococcus mutans. This bacteria can generate a huge amount of acid, which demineralizes the tooth and starts generating a cavity. In this cavity, if it's not stopped, it can generate pain, even tooth loss. So when there's a shift between a more stable biofilm dominated by Streptococcus sanguinis and some good strains towards these acid-producing Streptococcus mutans is where we get the disease. Now, interestingly, once bacteria can penetrate the outside layers of the tooth, they can reach dentin. Dentin is a tissue very similar to bone that also has an important collagen organic matrix in it. So once the caries is inside this dentin, collagen becomes a very important surface for attachment and bioinformation. And of course, therefore, these oral streptococci can actually attach the collagen via these collagen binding proteins that they can express on the surface, these proteins that allow them to attach to collagen. Now, collagen itself in the body, also in the tooth, suffers from certain changes with age, with the aging process, including the accumulation of advanced glycation end products, AGEs. One of the compounds that does this is methylglyoxal, and we know that this is an important factor in the elderly. And the elderly also suffer from specific caries that are very strong in dentin. So our question is, does MGO, the MGO AGE accumulation in this organic matrix or does this increase the attachment of Streptococcus mutans onto the collagen in the dentin surface? So the aim of this work was to characterize the impact of MGO-related AGE formation in collagen on the early attachment of Streptococcus mutans onto the surface. And to do so, we've used a combination, a very interdisciplinary approach, involving also AFM with living bacteria. So we have this in vitro collagen model, collagen gel model from rat-tail collagen that we can induce MGO AG accumulation with co-incubation with this compound. We can determine also how much AGs are being inserted into the collagen matrix with autofluorescence and also some microscopy characterization of this surface. And then to actually study how the bacteria attach this to the surface in real time with living bacteria, we've used some previously published protocols by other groups as well as our own group, where we can actually attach, first functionalize these AFM tips with polylysine that allows us then to immobilize living bacteria onto the tip of the AFM. So now we can put the bacteria in contact with the collagen in different states, and we can measure the unbinding forces between the bacteria. Again, in PBS, this is done with living bacteria, and we can obtain force curves. And these force curves give us a lot of information, but for purposes of the time I have today, I'll just focus mostly on the adhesion work that we can obtain from the area under the curve, on the adhesion force of these tiny unbinding events that we can see that we know are characteristic from the unbinding of specific adhesins, in this case, collagen binding proteins on the bacterium in comparison with the surface. And also we're going to look at the length at which these unbinding events are actually occurring. So regarding our model, or collagen model, we can characterize this with SEM and AFM. We can see that the native collagen has very individualized collagen fibers, not much bunching. That, of course, happens strongly once we start involving these advanced glycation end products into our matrix. And we can also see this with AFM 3D height images. Now, we've also gone to do some specific, nanodentation on these collagen fibers, and we can extract Young's modulus using, in this case, a DMT model. And we can see that there's an interesting increase in the Young's modulus of the cross-linked collagen, which is, again, consistent with previous reports and literature for different tissues, both in vitro as well as in vivo tissues. Now we can also quantify the way we can add these AGEs into the tissue with different concentrations of MgO over the course of 7 days. I'm showing here, we can see there's an interesting increase in the autofluorescence of collagen, which indicates in this case the accumulation of these products. And also if we add an inhibitor of cross-link formation of glycation, which is aminoguanidine, we can also see that we inhibit this process. So we clearly have a very simplified aging model in collagen that we can use in vitro to now look at the impact of this on bacterial attachment. So what we actually found is that these MgO-AGEs promote Streptococcus mutans attachment to the surface. We can actually see, if we look at the adhesion work in the cell, which is more reflective of the overall force between the cell and the surface, we see that this increases very interestingly when we have induced AGE formation in the collagen matrix. Now also, if we look at the specific peaks here on the force curve, which are reflective of the specific adhesion, in this case collagen binding protein interaction with the collagen, we see that the force of this interaction increases significantly in comparison with the interaction against the native collagen surface. And also, I mean, we can also plot this to see this in another way, the adhesion force versus the length at which this is recorded compared to the beginning of the unbinding process. And we can actually see that for the control state, most of the unbinding events, these adhesion events occur at a length underneath 500 nanometers, but mostly at the lower force range. And when we do the same experiments with the same bacterial probes, living bacterial probes, onto the modified collagen, we can actually see that this increased significantly. Now also, most of the effects are occurring, or most of these events are being recorded underneath 500 nanometers, although in some cases, specifically with the MgO surface, they tend to also become a bit more large. So in conclusion, we've seen that we have this in vitro aging model where we can show that the increase in MgO-derived AGEs increases the early attachment of Streptococcus mutans to the surface of collagen. Now, we believe that this is a combination of both cellular and subcellular events. So there's an increase in the overall adhesion work involving that the whole cell is having more interaction, but also the specific interaction, the specific collagen binding protein collagen interaction seems to also have a stronger force in this case. And we also see that most of these events, most of these interactions are occurring at distances below 500 nanometers, which is very relevant because most of the collagen binding proteins described for Streptococcus mutans in this particular strain that we're using are within this size range. So now we're going to use some molecular techniques to try to prove this at a molecular level, as well as also using some contour length modeling to look at maybe differences in adhesion involvement. The idea behind all this, of course, we believe and we hope that understanding this can improve our understanding of caries disease in the elderly, which is very associated to colonizing dentin, and maybe figure out new ways to prevent or diagnose caries in the elderly with more, in a more personalized medicine way. And also future work, we want to look at the interaction between Streptococcus sanguinis, the health-associated strain, with this altered collagen, in order to see how or if this collagen modification can also shift the ecology of the surface and promote this dysbiotic biofilm within the context of dental caries. So I'd just like to thank all my collaborators and the people that have worked on this, both in Chile as well as abroad, and also my early career research grant that has sponsored this. So I'd just like to thank you for your attention, invite you, if you're interested in doing some work abroad, maybe to have a look at our university and at our country if you want to have some sort of visit. So thank you very much for your time.
Thank you, Sebastian. That was a fab talk. Again, please pop any questions in the chat, but I mean, I already have lots of questions. You really reversed the paradigm there and put the bacteria on the probe. What made you choose to put the bacteria on the probe rather than the collagen?
So basically, this comes from the work we were doing during my PhD over there in London, and basically we found that it allows us to. It's easier to get the geometry, the bacteria itself, size-wise. Once you attach it to the tip, you're allowed to actually get a more specific and reproducible interaction with the surface. In other cases, when we've used, for example, yeast cells that can be expressed as a yeast, as a rounded cell or a hyphae that changes their phenotype a lot, we actually attach the surface to the tip and then use the immobilized cells on the surface. So in this case, since the bacteria are very reproducible in terms of the size, and we've been able to finally, after many years, get a more reliable way of immobilizing them onto the tip, we think that it's an easier for reproducibility.
And have you been able to characterize how consistent the bacterial layer is on the tip?
So yes, I mean, this is some work that we've done, and also many other groups are also doing similar work. Basically, the way we do this is that we use the AFM as well as a micro manipulator in a way, so we can actually get a dilution of bacteria that is not so concentrated, and we can basically see with the machine, we can approach a tip to some bacteria on a glass slide, and we can kind of see how we attach a tiny group of bacteria onto the tip. And then the measurements across different probes are very reproducible. So it is believed within this technique as well that the size of the contact at the end between the tip and the surface is usually around the range of maybe a bacteria, maybe a couple tiny cells, but it's quite reproducible across different probes.
It's very impressive. Heba has a question in the chat as well. Have you considered measuring structural changes in the collagen network?
Yes, that's a very good question. Actually, we're trying to maybe take this. I mean, I'm a dentist, so all of this, the big characterization part of things, sometimes is a bit over my head, but collaborations with Laurent Bozek, which is now in Canada, we actually want to maybe look at this also at a synchrotron level to try to use some infrared to see different changes in the collagen fibril that can be more associated to the collagen. But we also use, for example, molecular techniques such as ELISA, and we can also see the addition of these MGO-specific crosslinks into the collagen matrix. And again, we've done some nanoindentation with the AFM as well that shows an increase in the elastic modulus, for example, associated to the incorporation of these AGEs into the matrix.
It's very cool. I'm going to stop questioning you there. There's no more questions in the chat. So if anyone has extra questions for our two contributed speakers, I'm sure they won't mind you popping a message directly to them. Thank you both. They were really good. And now I'd like to introduce our final speaker of the day, our early career keynote speaker, and it's really with great pleasure that I'm introducing him. He is a keynote invited talk because he was the winner of the 2021 RMS Early Career Award, our very first cohort of that award. It's Kevin Whitley. He is based at Newcastle University, but his postdoctoral work has been admirably collaborative, split between Newcastle and Delft University of Technology. And he's now going to present the fruits of this work with a talk entitled Combining Nanofabrication, Microfluidics and Microscopy to Investigate the Dynamics and Functions of the Bacterial Cytoskeleton in Bacterial Cell Division. So over to you, Kevin, if you could share your screen.
Hi, there we are, right?
Yep, perfect.
Right. Yeah, so thanks so much for that introduction. I mean, it's wonderful. I'm really honored to be given this award. So to start out, I'd like to just give you the kind of personal background of how I got involved in this project. So I spent my PhD doing single molecule biophysics in vitro. So I was using optical tweezers to play with DNA and such, right? And so for my postdoc, I wanted to take this a step further and say, okay, let's look at how single molecules are going to behave in living cells, right? And I was especially interested in looking at bacterial cell division, which I'll mention why in a moment. And so I was looking around for postdocs, and I found this position that was being advertised between these two groups, K. Stecker, Delft, and Seamus in Newcastle. And the goal of which was to find a postdoc that worked between both groups and combine the expertise of both of them, so like nanofab and advanced microscopy, to tackle this subject. And so, of course, I couldn't pass that up. That was the subject I wanted to do, and involved this heavy method development aspect and new microscopy methods. So here I am. So advanced slide. Go. There we go. All right. So why are we interested in studying bacterial cell division? Well, there's basically two main reasons, at least for me, why I think this is important. One of which is it's a major target for current and probably future antibiotics, so think penicillin. because antimicrobial resistance is, of course, on the rise and is predicted, if nothing is done, to outpace the number of deaths from other causes within a few decades. So we urgently need new antibiotics. And from another more, I guess you could say, philosophical aspect or basic biology, there's this effort to see if we can build a cell from the bottom up, basically just take some whatever minimal components we can, some DNA, some proteins, some lipids, and see if we can get that to grow like a living cell and even to divide and reproduce. And so, of course, we would like to understand how simple, relatively simple organisms like bacteria do this so that we can inform that. And so, you know, for a lot of you here, I guess many of you are not biologists, right? Or microbiologists. So you might think, why are we interested in bacterial division in the first place? Like, isn't this a solved problem, basically? But it turns out it's actually a really complicated problem. So here's a model organism that I'm going to be showing throughout this talk, Bacillus subtilis. It's a rod-shaped organism surrounded by this thick cell wall. But so the physical problems that this cell has to solve in order to divide are actually pretty remarkable. I mean, it's under pretty high turgor pressure pushing outward, and it's surrounded by this rigid mesh of peptidoglycan, which is a polymer surrounding the cell. And so it's against this pressure and against all of this rigid polymer that it has to continually remodel and build further inward in order to divide. And at a molecular level, this is also a very complicated biophysical puzzle that needs to be solved, because there's a collection of a bunch of proteins that are going to work together and cooperate to build this cell wall further inward, this cross wall or septum. But each protein is on the order of, say, 5 to 10 nanometers in size, thereabouts, and they have to work together to build this ordered septum over a distance of about a micron, so it's about 100 times larger than they are. And so the collection of all proteins that are involved in doing division is called the divisome. So, I mean, fun fact, anything in biology is like 10 times cooler sounding if you add "ome" to the end of it, and so that's why this term exists. Anyway, but so divisome proteins vary a lot from species to species, and there's still tons of work that still needs to be done to understand the functions of each of these and such. But thankfully, there are a few shared components across most bacteria, one of which is the synthases that actually build the cell wall, the PG synthases, because of course they are going to need to be there. But another is this protein FTSZ. And so this is a cytoskeletal protein, so you could think like microtubules in eukaryotes. It's actually a homologue of that. And this protein is important because it's almost universally conserved throughout bacteria and essential for division in almost all. There's a few exceptions, but by and large, it's very widely conserved and essential. And so because of that, it has been the subject of intense research over the past 3-ish decades or so. But I'm going to skip ahead to 2017 to some results that our labs contributed to prior to my arrival. So 7 groups were working together actually on this subject in different species. And they looked and found that FTSZ, even though it's the skeleton of the cell, it's highly dynamic. It actually moves circumferentially around the cell, and in doing so likely guides the synthases to build the cell wall. But it's an unusual kind of motion. So I'll play this animation. It's called treadmilling. And so this is where the filament center of mass does move. There's this collective motion, but actually each individual monomer doesn't. Each one just kind of binds onto one end, sits there for a while, and then eventually falls off the other end. But the filament itself does move. And so this is called treadmilling. But despite these advances, it's still unclear basically what the physiological role of FTSZ treadmilling in the cell is, despite how important it is. So I'm just going to show you what the conclusions are right from the start, and then we'll go into especially more the microscopy and the method development, because of course this is a microscopy congress, right? So anyway, with FTSZ, we found that it actually has 2 roles, one of which is to basically set up the FTSZ ring, the division ring, and prepare it for division, and afterward to guide the synthases. But there is a point at which it does become non-essential, so you can just stop all FTSZ motion, and cells are still going to keep dividing, which does have some implications for antibiotic design as well as for informing the bottom-up biology. Right. So we wanted to look at FTSZ treadmilling across the whole cell cycle to see really what its point is. But of course, that raised the question: what are the stages of division? How does division happen in bacteria? So we looked at this in terms of the large macroscopic structures in the cell. So this is a video here of Bacillus subtilis cells. There's, I guess, a couple of them in this field of view, well, one then two, where FTSZ is labeled with a fluorescent protein. So we are just looking at the larger scale changes in the cell over time. So we quantified this with some tracking software as well as some MATLAB software that we wrote to quantify these larger scale changes in the cell, just its diameter and its thickness. So here's just some traces showing that, of course, the black lines are representative, whereas the gray dotted lines are many others, diameter and thickness over time. And so from these time traces, we can identify 3 stages for the cell division. One of which is what we call the nascent ring stage, where it's still not even really a ring at this point. It's kind of a diffuse collection of filaments, very blob-like, that you can see in the video. But then after that, this condenses into a tight, dense Z ring, which we call the mature ring, before it has actually begun constricting. And then after that, it is now constricting. The diameter is decreasing. So, okay, we wanted to look at FTSZ treadmilling at each of these stages to see what's going on. But doing, molecular level imaging in bacteria is difficult for a few reasons. One of which, little known fact, bacteria are small, okay? And I mean, they're smaller, much smaller than eukaryotic cells, which is often, I think, maybe not quite as appreciated as it could be. But they're not that much bigger than the diffraction limit of visible light, right? And so if you just put some single molecule in there and you image it, well, I mean, you're going to get a lot of signal overlap if you have more than one, especially if you're looking along the short axis of the cell, which makes this actually pretty difficult to image. Another fact that complicates this is that a lot of bacterial cells are rod-shaped, especially most model species and a lot of human pathogens are rod-shaped. And so when you image them on a microscope, they lie flat, so they're horizontal, and the division ring, you view it side on. So it's projected into a line, essentially, on the cover slip, and you see substantial signal overlap. And you can get around that with using total internal reflection fluorescence, where you only illuminate the bottom, say, 100 nanometers or so of the cell. So you remove a lot of these out of focus filaments, but then you also lose the context of the entire cell. So, I mean, this is what you get from that. You can get quantitative information out of it, although it is actually very difficult. And I can say from analyzing some of these images, it is really difficult to get information out of that. So then, wouldn't it be great if instead of having the cells lying flat on the microscope cover slip, you just stand them upright? You just put them in tiny little holes that are the shape of bacteria, and then you have the division ring in a single imaging plane, so you have higher resolution and no signal overlap. And so what we do is we use nanofabrication techniques in a clean room, e-beam lithography and such, to make micro pillars on a silicon wafer to use as a mold, and use those as a mold, pour on some molten agarose, let that set, and then you have some bacteria-sized holes that you can stick the bacteria into, so they're all stood up, and you can see them all in, a nice, neat array in your, field of view. And so in doing so, we can see FTSZ moving around the circumference of the cell in pretty high resolution without even messing with any, fancy fluorophores or anything like that. Actually, I'm going to replay this because it's a cool video. Wait, hey, you're supposed to play. Anyway, so we are now calling this technique Vercini for vertical cell illumination by nanostructured immobilization. And even though this technique is really good resolution, we found that it's actually not even quite enough for looking at nascent Z rings because they're so dispersed and so weak in signal that it's really hard to see anything. So we had to adapt and come up with some image processing methods to fix this. And this video really doesn't like to replay, so I'm just going to have to do this manually. So anyway, we denoise the images first, and then we use an explicit model to subtract out the cytoplasmic background. And so in doing so, with Vercini, with this image processing, we can get the highest sensitivity FTSZ treadmilling that has been done to date. And how we analyze these videos is we look at treadmilling for some time. We fit the cell to a circle, and we produce a kymograph, which is radial kymograph. So this goes from 0 to 360 degrees around the cell, and then a second time around to 720 degrees. So it's plotted side by side. And we do that just so we can see this boundary between 0 and 360 degrees much more easily. So how to interpret these kymographs? Imagine if you had, say, a stationary particle that's just stuck at 180 degrees around the ring. So over time, if it just sits there, you're going to have this vertical line at 180 degrees. Whereas if you had directed motion, if you had something going from 0 to 360 degrees around, you're going to end up with some diagonal line. And so what you see in this example kymograph here is a lot of diagonal lines moving in both directions, because FTSZ is treadmilling bidirectionally around the cell. Right. So then coming back to this point, we wanted to look at how this treadmilling behavior is happening across all these different stages of division that I mentioned before. Nascent, mature, and constricting. So we've done this for a bunch of different cells now. So the mature and constricting rings look pretty similar to the example I showed you before, with this kind of almost lattice-looking diagonal lines crisscrossing and all that, consistent treadmilling. Nascent rings are much more sparse, and they have a lot of filaments that aren't really moving much. They're just kind of sitting there. So of course we've done this for many cells, and this is just a summary in the violin plot saying what I just said. Nascent rings, a lot of really static filaments. Some are moving variable speed. Mature and constricting rings, very consistent motion, actually. Right, so that was pretty cool, but we wanted to take this a step further. And we wanted to use specific antibiotics to perturb the cell and see what's going to happen to the system in that case. But the problem is, with this original implementation of the method, we have this block of agarose sitting on top of the cells. And so it's not very easy to control the chemical environment because it has to diffuse through this porous medium. So the idea then is, if we just invert the system and we make it so the holes are open on top, and we put the whole thing into a microfluidic chamber, then we can more accurately control the chemical environment and flush over different nutrients or perturbatives like antibiotics. So this works basically the same way. We have our silicon pillars as a master, pour on some PDMS, which some of you might know it's commonly used for microfluidics, and then peel that off, and we have a cover glass with PDMS on it, and then we get the cells into the holes, which it turns out is not a trivial matter. We actually centrifuge them into the holes by spotting them on top of the holes and sticking the whole thing into a centrifuge and just spinning them in. But so at the end of this, we have a microscope cover slip that has the cells loaded into the holes that are facing up, right? So what we want to do then is put this into a microfluidic chamber so that we can actually flush over perturbatives and such. So from this, I took a page from my single molecule background and made some low-tech flow chamber. And so this is a microscope cover slide that has some holes drilled in it, some pipette tips inserted in and epoxied in place, and a flow channel that's made out of a piece of double-sided tape, which is about 100 microns in thickness or something like that. And I found that actually the brand of tape really matters. I use duct tape. I found out that Scotch doesn't really work. It doesn't stick to PDMS super well. Anyway, minor point. But so we assemble this whole thing, and we have this nice low-tech, low-cost microfluidic chamber over which we can flow stuff and affect the cells. And we're now calling this technique mu-Vercini for microfluidic Vercini, remember? Okay, so we set out to test this method as well as to gain some biological insight by using this antibiotic called PC19. So this compound specifically inhibits FTSZ. And how it does that is it actually binds to it and it prevents it from depolymerizing. So it's unable to move then. It's similar to Taxol for microtubules, if you're aware of that. It's an anti-cancer drug. But it still remains unknown up to this point what the timescale of effect is and if it really works well against rings that are in the middle of constriction if they've already, started going. So using mu-Vercini, we record the dynamics of FTSZ treadmilling for a while. This looks the same as the things I showed you before. But then, of course, we flush in this compound and things go out of focus for a few seconds, hence the black band there. But then after this compound hits the cells, within a matter of a couple of seconds, basically, all motion stops. FTSZ treadmilling is halted, and you just see these static filaments afterward. So it is actually a very potent compound, and it works very well against all these stages of constriction that I showed before. So we then wanted to see what's going to happen when we stop FTSZ treadmilling abruptly in rings that are in the middle of trying to condense their ring or trying to constrict. And in this case, we just used horizontal cells because it turned out to be faster and easier, and we didn't need super high resolution for it. So recording the cells growing, dividing for a while, flush in PC19, and you can see this very characteristic aspect of PC19 where FTSZ clumps form when they can't depolymerize anymore. It's a very dynamic process. But so, of course, from these videos, we wanted to try and see what is this doing to the condensation of the Z ring and also to actively constricting rings. And so if we look at a bunch of nascent rings, which of course aren't really rings, it's almost a misnomer there, after PC19 hits, you see basically nothing. I mean, there's no ring or anything that forms, it's just blobs of junk. I mean, they never condense. And I gave a few the benefit of the doubt because they maybe did. But for the most part, none of these rings are able to condense after this compound hits them, telling us that FTSZ does need to be treadmilling in order to condense, probably just to interact with other filaments that are moving around. Then looking at constriction, we wanted to see if this was going to affect how the cells are constricting when we inhibit FTSZ treadmilling. Nascent rings, of course, don't. I mean, they don't even form a ring, so, move past those. For mature rings, we see two different behaviors. Some stop and they don't constrict at all, and some do, actually, even after the compound hits them. And for rings that are in the middle of constriction when this compound arrives, they continue constricting pretty much unaffected. And that's summarized in this stacked bar plot here. And we went a bit further than this to show that actually it's the arrival of the PG synthases that are building the cell wall inward that corresponds to when FTSZ treadmilling becomes non-essential. So when those enzymes arrive, you don't need FTSZ around anymore, apparently, or at least not to move. But this was a surprising finding to us because we, of course, know that FTSZ is still dynamic in constricting rings. It is still moving, definitely. And we know that it does, to some extent, affect the rate of constriction. So we wanted to more sensitively look at rings during constriction when FTSZ treadmilling has been knocked out. So we tracked these cells, we measured their diameters with our MATLAB software over time, right? And so this is the diameter of the cells over time, they're constricting, right? And we fit these to a very simple toy model for constriction where all it really assumes is that the rate of cell wall insertion going inward is constant. And so that just begins at time t equals 0 there. And so from this, we can get the rate of constriction as well as the time that it takes to constrict, extrapolating from 0 to the end there. And comparing that from the cells that have active Z treadmilling to those that have it knocked out, those that are treated, the ones that have been treated constrict considerably more slowly. And I mean, after eventually finding this, I could look back to the videos and be like, I guess they are actually slower. Interesting. So it's slower, but the cells are still able to divide even after treatment with this compound. And so then what's going on with this? So our model for this is that FTSZ treadmilling is needed basically to confer long distance order to the PG synthases. Like I said, these synthases are 5-10 nanometers in size. I mean, they don't have any directional vector telling them where they need to go. But FTSZ is forming these filaments that's able to move. So that might be actually giving them that directionality that they need. But then after you have a septum that's already started, it's already started building one inward, the synthases already have a track to move on, so to speak, and maybe they don't really need FTSZ to guide them anymore. But in the same sense, maybe there's some effect. So if we look at a few cases that I've found, when the cells are treated, sometimes the dividing septa go very strange and off-axis, like diagonal, which may be, an indication of this. This has also been seen in previous SEM images where the septa end up really messed up looking. after they've been treated. So the cells are still able to divide. They just end up with a really messed up looking septum, which is probably not very happy. Anyway, so this comes back to the final conclusions for the FTSZ treadmilling stuff. That you do need FTSZ treadmilling to condense the ring. Probably just treadmilling filaments are moving around the circumference and then running into each other. And then other factors are bundling them together and forming this nice dense ring. And then after that, they're probably guiding the synthases by giving them some longer distance order. But of course, crucially, after the septum has begun, you don't really need it anymore, apparently, which also says that, if you're designing antibiotics against FTSZ, which would make sense because it's essential for division, there is a subset of cells this is not really going to be working on, those that have already started division. And also this is helpful for, say, the minimal cell effort, trying to build a cell from the bottom up, that you might not want to use just FTSZ, at least, to divide the cell, because it seems like, at least in bacteria, they need quite a few other components, and that FTSZ itself is not the only thing you need. Right. But so then I have just a few more minutes. I'll just explain kind of how I'm following this up at the moment, because I find it interesting. Right. So I mentioned that these PG synthases are the ones that are moving and doing all the actual work of building the septum. And so that's what I'm trying to do now, is to look at the synthases moving it at single molecule resolution. So I'm doing single particle tracking on these synthases while the cells are confined vertically in using our Vercini method, and watching these single molecules moving around the septum, probably doing their thing, building the wall. And in this case, we have a single molecule that we're tracking around the septum for close to 5 minutes there in that case. And I'm hoping to get even better data in the future, or at least a lot more that look like this, because these are very nice tracks to see. And you can see a lot of this rich behavior in the distance that it's moving, that there are these direction changes, there's pauses. I mean, there's a lot of rich behavior that we have to look at, which I'm really excited to get deeper into this project. Yeah, so for my last slide here is basically just an advertisement for Vercini. So you don't have to have rod-shaped cells for this. Any cells with cylindrical symmetry, including Streptococcus, like we heard about in the last talk, will fit in these holes that you can image. And you also don't even have to image division proteins with these. We found that you can also image the cell poles with them really well. In fact, because the cells are adjacent to the cover slip, you can use turf, like I mentioned before, to only illuminate the very bottom 100 nanometers or so of the cell. And you can see the poles of the cell in, with high signal to noise ratio. So here's an example of a strain that we got from Henrik Strahl here in Newcastle, which is a chemoreceptor protein. And you can see that in horizontal cells, it's really hard to see much of anything that's going on. But if you orient them vertically, you can see these large chemoreceptor clusters, which are extremely immobile, as it turns out. But we think that you could do quite a lot more with this. So, I mean, get in touch if you're interested. I mean, we are trying to democratize this method. We have this new preprint out, and we're in talks with a company to try and make these silicon wafers that we can actually, distribute to other groups. No proceeds come to us, but yeah, we hope that you'll find this method interesting if you're doing bacteria and such. And so obviously all this is a team effort, and a large team indeed. So I'd like to acknowledge all the people who've contributed to this, both in the Holden Lab and in Kay Stecker's group, now solely based in Seamus's group, but of course, I acknowledge the work that was done, in both groups and everyone that helped out along the way. A special shout out to Callum Jukes there in the X-Men shirt, who is I think it's X-Men anyway, who's a PhD student in Seamus's group who contributed quite a bit to what I presented today. And one last thing is, I'm now looking for PI level positions. So, hey, if you're a department that is looking for a microscopist, get in touch. I'd love to hear from you. And so with that, I will finish. Thank you all for listening. Thanks.
Thanks, Kevin. That was a great talk, and I enjoyed the little advert for yourself at the end there as well. I hope someone comes back to you. Please, people, feel free to pop a question in the chat. I'm sure there's lots of questions coming. In the meantime, I was wondering a bit about the distance from the objective to the that actual ring, when you're sitting them upright, it must affect the imaging a little bit, or the focus working distance of the objective that you need.
Well, at least in the case of the original Vercini technique, it's about 1 or 2 microns off the surface. So that one's not so bad. We use a 100x turf objective for that one. And I mean, we have pretty high resolution, as you can see. With the other method, mu Vercini, where we have the PDMS layer on top of the cover slip, that makes it much more difficult. And we found that with the 100x turf objective, we can't actually image high enough because the working distance limits it. So in that case, we use a silicone oil immersion objective, which is for imaging into deep tissues, I guess, is the reason why it was made. But that enables us to get much higher working distance to image all the way up there.
Yeah, and do you get much signal loss having to get at that extra distance?
It doesn't seem like it. I haven't quantified the loss of signal, but yeah, it seems pretty similar, at least in both methods. So, yeah.
And how about the imaging pores when you're treating the cells and they're extending? Because we could see in the horizontal phase that they were extending quite rapidly. Do they extend out of the pores? How does that work?
Yeah, they do actually. And so if you're doing this in, say, fast growth conditions, sometimes you have to do the imaging a bit quickly because they will start to grow out over time. And that's one of the reasons actually we were doing the constriction work in horizontal cells, is because we did find that cells were growing out of the holes. Actually, I have a slide for this. There we go. So we were imaging constriction in these open top microholes initially, but the problem we had was that over time, a lot of these cells are just popping out of the holes. They are just growing out over time. So this is probably a surmountable issue, but we haven't devoted enough time yet to trying to find a way to really glue the cells into the holes to make sure they don't grow out.
Sure. Okay, I asked a predictable question there. I've also got a question from Liam. He's asking, how picky do you need to be when tagging the FTSZ to retain full functionality, and are there any particular tags to avoid?
So with FTSZ, it is just a tricky protein to tag in general because it is so essential and it interacts with a lot of other things. So this strain we're using dates back, over 20 years actually, because it's an unusually robust FTSZ fusion protein that is actually fully labeled. You can also express this at dilute levels, and in that case you can use a number of other labels like M-neon green and things like that, but we've chosen to use this FTSZ GFP strain mostly because it doesn't really perturb the cells as much as most other tags do.
Okay. And I've had another question about the inhibition. Have you tried reversing it or removing the inhibitor, and do you see the rotation movement begin again?
I have, yeah. Early on, I tried doing those experiments, these kind of wash out experiments, as I called them. And yeah, I think I was able to see some restarting of treadmilling, but honestly, it's really difficult to see because I think this particular compound does stick fairly well into the FTSZ. So, I have to try and image. What I was doing is imaging, for a while, waiting 10 minutes, coming back, trying to image it again, because I think FTSZ does restart treadmilling after you wash this out, but it probably takes a little while to really get to that point.
Okay, it sounds like quite a robust inhibitor and the timescales that it works.
Yeah.
Yeah. Do you think the agarose as well maybe absorbs some of it? I don't know how embedding in agarose could.
Well, at least in the case of the when treating with the compound, there was no agarose in that case. That was with PDMS. We have looked at a few other inhibitors, though. There's one that had been developed called PC58, another kind of experimental antibiotic. That one works the other way by preventing FTSZ from polymerizing in the first case. So we've also looked at that one. Unfortunately, it didn't quite have the shocking effect that we were hoping, because the constricting rings did not completely dissolve when we treat with this compound. They're pretty well stuck together, unfortunately.
Sure. Okay, well, thanks so much for answering all those questions and this fantastic talk. Thanks for also applying as our first medal winner. I'll let you off the hook now and hand back over to Liam, who's going to kick off the AGM section of the meeting.
Super, thank you, Becky. Yeah, that was a great talk, Kevin. Thank you very much. And thank you to our other speakers in that session as well. So yeah, this next part of the meeting involves the section AGM for the Early Career Committee. But first of all, I would like to announce the winner of the Best Presentation Award for our contributed talks. So the committee have been discussing things in the background via WhatsApp, and we've agreed on a winner for that award. So we'd like to congratulate Sebastian Agayo as the winner of Best Talk today for our contributed speakers. I don't know if Sebastian, if you're still on the call, if you want to give us a wave so we can see you. There you are. Hello. Yes, congratulations, Sebastian. Well done. It was a really great talk. A lot of different interdisciplinary techniques there, and something for everyone, really. The RMS office will be in touch to arrange your prize, and we'll get you a certificate and stuff. So yeah, really well done. That's some cool stuff there. And thank you for joining us all the way from Chile. What time is it in Chile?
It's 11:30 in the morning. It's fine. We're GMT -4 now, so it's a very reasonable time. Thank you so much. I really appreciate it.
Thanks, Sebastian. Cheers. Okay, so that brings me on to the final part of today. So I'm just going to kind of share the agenda for this AGM. It's as I say, this is our first RMS Early Career AGM, so it's going to be a kind of informal, very quick, and there will be an kind of open discussion at the end of it. I'll just stay on the call, and if anybody wants to chip in on how we can improve what we're doing, or if you think we're doing anything very well, you can tell us. And yeah, I'll just hang around in the call, and after I've gone through the meeting, then people are free to drop out as they go. So we have no matters arising because this is our first AGM meeting, and my report will give you a brief update on everything that we've done this year. So this has been my first year as AGM, as chair of the Early Career Committee, and it's been an enjoyable year for the committee. We've achieved a lot of great things in an otherwise turbulent time. Before summarising this year's highlights, though, I'd like to thank the previous chair and founder of this section, Dr. Colm O'Leary, who's now settled into his new role as a postdoc in California. Colm really shaped the Early Career Committee into what it is today, and has brought together a fantastic group of early career microscopists to serve the interests of the imaging community. And this has made my job a lot easier as chair by having such a great group of people to bounce ideas off of and really drive the section forward. The remit of the Early Career Committee is to provide a platform for early career microscopists with the full support of the RMS, and we aim to provide an inclusive environment for early career researchers to showcase their research, discuss their roles in the context of the field, and to help disseminate and develop resources for early career scientists. Any students or postdocs or early career professionals who are members of the RMS can benefit from our activities. And the best way to keep up to date with our activities is primarily through our Twitter account, which is at RMS_early-career, or by the RMS website, emails, or features in InFocus magazine. We began the year by setting up our official Early Career Twitter account, which we use to communicate to our members. This has been a great success, and it's been driven by Dr. Emma McDermott, who you met earlier, and is now seen to an increase of over 500 followers, with daily content on job posts, career resources, events news, and technical overviews. The section has also seen the launch of the inaugural RMS Early Career Award, which is a prestigious prize awarded to an outstanding early career microscopist who has pushed the boundary of their field and actively communicated the impact of their work. I'd like to thank those on the committee for their work in pulling this award together, and for all of the outstanding submissions that we received for that award. There was some really great science in there, and I really thank all of the applicants for that. Our recipient of the 2021 RMS Early Career Award is Dr. Kevin Whitley, as we all know, and he provided a really excellent overview just there of his research. And Kevin will be formally awarded and commended with his award at an in-person RMS event later in the year. We're happy to also announce that the Early Career Award will become an annual feature of the committee calendar, and it will be presented in line with other RMS section awards. The committee have been working closely with Doctors Georgina Fletcher, Nicola Lawrence and Alex Sosic to develop and launch a series of short technical lectures, which are named Technical Tea Breaks. These 15-minute long videos have been created by experts in their field, and some are publicly available on YouTube currently. These are currently limited to light microscopy, but we hope to extend this into EM and image analysis in the coming months. The committee have been actively engaging in developing regular features for In Focus magazine, and these features cover committee membership introductions, updates, events, news, perspectives, career spotlights, and more. And we look forward to furthering our engagement with the In Focus team over the next year and continuing promotion of our RMS Early Career members. The committee has been implementing its official constitution, which brings it in line with other RMS science sections. And this effectively formalizes the committee membership. And I'm pleased to say that everyone has agreed to stay in office for at least one more year. And I look forward to working with you all over the next year and continuing to do great science and great initiatives. The major event for the committee's calendar has been today's Early Career Symposium at mmc 2021. So I'd like to take this time to thank all of the committee members, the session chairs, and the RMS team who've helped to organise this event. Also extend a thank to our panelists, our sponsors, our selected presenters, and our keynote speakers for their involvement as well. We've also been heavily involved in organising a virtual social event for mmc 2021, and we've partnered with Hotel Chocolat to provide a bespoke tasting event. I'm very grateful to Communications Physics and their generous sponsorship of the event, which has allowed us to heavily subsidise ticket prices and make the most out of effectively sitting at home eating chocolate when we should otherwise have been partaking in post-conference in-person extracurricular activities. Looking ahead, the committee is in really good shape. We will continue to serve the best interests of the community in the next year. We're currently working on developing specific early career content for the Journal of Microscopy and developing future ECR-focused events. Moreover, we look forward to continuing the RMS Early Career Award into 2022 and showcasing the fantastic research of our membership. Finally, I would like to thank Miss Alison Winton and Miss Amanda Jarman and the rest of the RMS team for all of their hard work and commitment and dedication to the committee, which has really driven and supported a lot of the efforts behind the scenes. So that brings me to the end of my update report. There are no nominations to committee to report, and I'm delighted that everybody has agreed to stay in office for their next term, which is effectively 1 year from now. The full details of the committee, including a transcript of everything I've just said, will be made available in due course online at the Early Career page on the RMS website. If there's anything else that we can kind of bring up, then I'll open the floor in a minute or so. All I'd like to say is thank you, everybody. I hope you have a great MMC, and I hope you all take the time to go and check out the exhibition and trade show, which is free. If you've not registered for MMC, then registration is still open, so you can check out the science program and still register to attend any of the sessions that you want to. So yeah, I'll stop sharing my screen now, and I will stay on the call. I will open up the floor. Thank you all for attending, and yeah, we've had a really great session and some really good science. And I'll stop talking now so it's not awkward. Thank you, everyone.
I just wanted to thank everyone. This was a really fantastic event. Liam and the whole team, the Early Career Symposium was just fantastic. And I really enjoyed being involved and listening to all those great talks. So thank you so much again. You know, you're the future of RMS, and I'm just really, really thrilled and delighted to be here. So thank you all, and I hope you enjoy MMC 2021.
Thanks, Grace. Thank you.